Ooh, okay, cool. Oh, well, it's three.
Let me see if it's recording.
Yeah.
Check with Steve and make sure he's in here.
Is he on?
He's right.
Steve, can you hear all right?
Say Dr.
Yes, I think I'm good. I followed an old calendar item. It led me to Teams link.
Audio.
And pulled me out of the fire.
It was working when we were on the lab meeting Zoom.
So who can't hear?
We can't.
So Steve was just talking and
We couldn't hear him.
Yeah.
So tell everybody on Zoom to write in the chat their questions.
Okay, so
Or
yeah,
I can say that.
yeah, we can have.
And what we can maybe do is
Okay.
for private defense
Mhm.
time, we can put him on my computer or
Oh, yeah.
something if he needs
Sure.
to talk about it.
Sure.
Great.
Okay.
Are you ready?
Do you want to use this?
You can't hear me?
Then we can do a little clippy action. Okay.
Okay, good afternoon, everyone, and I hope all of you that are joining on Zoom can hear me.
Hi, Steve.
So for those of you who don't know me, I am Lori McMahon, and I have the honor of being Rosie Rae's dissertation mentor. So I'm going to share a few things with you about Rosie, but I think that any day that we graduate another PhD scientist is an amazing day. And so today is an amazing day because we have another PhD scientist who is finishing her dissertation today. With a caveat, she has to finish her private defense after this public defense, and I'll tell you more about that in a moment. So, and especially right now, I think we're all feeling a lot of extra anxiety based on what's going on with the federal government and funding for science, and for us, particularly with the National Institute of Health. All of us as biomedical scientists, we're all keenly aware of what's going on now. So that makes this day an even bigger day to celebrate. So Rosie grew up in Livonia, Michigan, and her family is here. So thank you so much to her parents and her friends for joining us today. And she started her research career very early as a freshman and undergrad. So she graduated from North Central College. Her undergrad degree was in molecular neuroscience with a double major in biology and chemistry. And when Rosie was an undergraduate, she received many research opportunities, fellowships, awards, including one that took her to England. So she spent a summer in England. She was a genetics fellow. She's done all kinds of work in neuroscience, but also starting one of her most early experiences was with bacteria. So she graduated with amazingly impressive credentials as an undergrad with her research experiences. And one of those research experiences too brought her to the University of Alabama at Birmingham with an NSF-funded REU fellowship. So that experience for that summer led her to apply to the University of Alabama at Birmingham for her PhD program. And that's where Rosie and I met. I was a professor of neuroscience at UAB and the dean of the graduate school, and Rosie applied to the Graduate Biomedical Sciences PhD program, an umbrella program in cellular, molecular, and developmental science. And she found my lab and did a rotation, and that was a great day for me when she approached me. We talked a lot while you were looking for rotations. And during her rotation is when I was recruited here at MUSC. And I wasn't sure if Rosie would want to pack up, because she had just moved about 9 months ago to Birmingham from Michigan. And luckily for me and for our lab, Rosie trusted me enough to pack up again and move here to MUSC. And it's been just an incredible honor for me to serve as her mentor. And just like undergrad, during her PhD training, she has been highly involved. She served as co-chair of the Neuroscience Retreat. She's been really active in the Alzheimer's Association for the International Conference. She's been co-chair of the Sex and Gender Increased Risk for Alzheimer's Disease, so they have a special initiatives in the Alzheimer's Association for that. She's led as co-chair and helped facilitate meeting planning. She was awarded a fellowship on, Rachel, awarded a fellowship from CTSA. So she was a TL1 fellow for a year. That paid her stipend, and she got to travel with that award too. But before I turn it over, I know I'm going long. Before I have to tell you the story about Rosie. She's heard me say it before. So my lab has done brain slice electrophysiology, and my current grant, we were funded to do recordings in vivo in the locus coeruleus. So all you neuroscientists in the room know locus coeruleus is really tiny and it's hard to target. And I was wondering who I would refer to the lab to work on this. Rosie, as our first year PhD student, decided to buy a 1970s minivan.
It's an 89.
89, okay. And she was in the process of converting it to a tiny house. So she would come to lab meeting and she would tell us stories about cutting a hole in the roof, and she was putting in an air conditioner, she was installing a refrigerator, she was going to this garage and learning how to do all this work from mechanics. I thought she can report in the local. No, yeah. With that persistence, perseverance, and just courage and guts. And that's what it takes to set up a vivo recording and in a lab that doesn't have that expertise. So she pioneered it in my lab, and I'll be forever grateful to you for that. So she's going to talk to you about those data today. But it was a lot of grit and perseverance and fortitude, everything that you need to have to be successful in this career path. And she did it by collaborating, and Steve Shea, one of her committee members who's on Zoom, is an expert in locus coeruleus recordings. He helped us, is still helping us. She's going to have 2 first author papers out of her work that are beautiful. And so you will hear about that today. So all of those attributes, her experience, her perseverance and drive, is what has gotten her here today. And I'm just been honored to be her mentor. And I'm going to quit talking because really it's her day. So Rosie, take it away. Thanks. Oh, and just to tell you, you can ask questions during her presentation, so don't be shy, raise your hand, ask questions.
Actually, I would prefer if you guys held them until the
Oh, do you want that?
Yeah, I
Okay.
do. I have
Alright,
slide
I
numbers
stand
on the slides, and so if you guys want to just like take a note of what slide it's on, and then yeah, we can answer all of them again.
Okay, and those of you who are on Zoom, you can put questions in the chat. Following her oral presentation and the Q&A, we will dismiss all of you. The committee will stay in this room for the private defense. And then, assuming all goes well, we will be back in this room for some refreshments to help celebrate it. Right, your way.
Thanks, Lori. That lovely introduction. I basically like, yeah, you can do this. I said, yeah, sure. And here we are 5 years later. But yeah, so but yeah, so my name's Rosie, and I'm going to be talking about what I've done looking at physiology of the locus coeruleus. So we'll start with the clicker working. Hold on.
All right. Hold on. Okay, so I'm going to start with a brief outline, yeah, okay, start with a brief outline of what I'm going to be talking about today. So first I want to start by talking about my favorite brain region, which is the locus coeruleus. Before I get into the physiology of it, I'm going to define some terms that I'll be using just so that we can all be on the same page with it. And then I'm going to get into the 2 projects that I worked on during my time here. The first was looking at binaural modulation of the physiology. And the second was looking at the effect of ovarian hormone deprivation on locus coeruleus physiology in this Alzheimer's disease rat model. And then at the end, I'm going to kind of integrate everything and talk about how this adds relevance to the field. And then throughout this presentation, I'll be using these little pink stars to kind of highlight things that are important or that I might be talking about later in the talk. So to start with the locus coeruleus, this is a tiny nucleus down in the brainstem. When I say tiny, it is about the size of a baby's fingernail. Now, despite this small size, it has these vast and expansive productions to majority of the brain, upwards of 90% of the brain. Now, the locus coeruleus
produces noradrenaline for the majority of the brain. Noradrenaline is similar to the compound adrenaline, it's only nothing different. So you can kind of think of this as neuroadrenaline, noradrenaline. Now, while adrenaline is hailed as the fight or flight molecule, I want you to think of noradrenaline as more of an arousal molecule. But more of an arousal molecule. And so because the locus coeruleus has all of these projections going everywhere in the brain, it really has its fingers in a ton of the fingers. So I put all of these up here just to underscore the importance of this tiny brain structure just in everyday basic biologic functions. I also want to note that it's been shown that the LC is dysfunctional in certain diseases. We'll be getting into this a little bit later. So starting with the anatomy of this structure, because it is so small, it has been historically thought to be a homogeneous population of noradrenergic cells. But there's been some really nice literature coming out more recently showing that there's actually a lot of heterogeneity. This has been shown morphologically, physiologically, and productionally as well. Now, the LC does not exist in a vacuum. Situated down in the brainstem, directly outside of it is this area called the pericoeruleus. Now, this is made up of non-noradrenergic cells, and these cells kind of reach into that nucleus to innervate and synapse onto these cells. So now onto the physiology terms that I'm going to be using. I've been talking about neurons. I'm also going to be calling them units. This is a product of the analysis that I use. Now, neurons are electrically active cells, so when they fire, it looks something like this, like a spike train. Now, one of these events is an action potential or a spike, as I'll be referring to it as. This is when a neuron fires. Now, by taking the amount of spikes that happen over a certain amount of time, you can calculate that neuron's firing rate. This is measured in hertz. You can also look at the time in between the spikes. This is referred to as the interspike interval, or the ISI. And then there are these bursting events that happen. Now, there are very specific criteria to define a locus coeruleus burst, but you can think of this as many spikes within a short amount of time. So now getting into the physiology of the locus coeruleus. So the LC has been reported to have two types of firing. The first is tonic firing. It looks something like this. It hits at about 1 to 2 hertz. And it's needed to maintain a baseline state of arousal. All of you sitting here, this is what your locus coeruleus is doing right now. This is why you're upright, paying attention. I'm very thankful for that. But just needed to maintain that baseline state of arousal. Now, the LC can also participate in what is called phasic firing, or that bursting firing. So this hits a lot higher, on the order of 10 to 20 hertz. You can see there's a lot more activity going on. And this is needed to increase noradrenaline to the rest of the brain. Now, this is typically in response to a stimulus, but this can also happen spontaneously. Now, the benefit of studying this burst firing is because the burst firing increases noradrenaline to the rest of the brain and helps an organism mount a arousal behavior, and these bursts have been closely tied to differences in cognition. So by studying these bursting events, we can gain insights into downstream noradrenaline flows. I also want to note that the balance between tonic and phasic firing in different diseases is dysregulated, and this is what's thought to contribute to those arousal-related symptoms that we see, things like anxiety, irritability, and sleep disturbances. Okay, so getting into the first project that I worked on during my time here looking at the diurnal modulation of locus coeruleus neurons. We're working on the reviews for the Journal of Neurophysiology with this, but it is currently available on bioRxiv, so if you want to check it out. So first I want to tell you about circadian rhythms of the locus coeruleus. Now, a circadian rhythm is a physical, mental, or behavioral change that an organism experiences over a 24-hour period. Not only is this happening at a top level, but this is happening in every single one of our cells in the body at a transcriptional and translational feedback loop that creates these time of day variations within our cells. Now, in individuals with Alzheimer's disease and dementia, there's this phenomenon that's called sundowning. And so this is where there's an increase in those arousal-related symptoms, anxiety, and irritability that happen later on in the day. And this is in individuals with Alzheimer's and dementia. And so considering the dysfunction that happens in the locus coeruleus and the locus coeruleus' role in these arousal-related symptoms, this begs the question, do these noradrenergic cells have a circadian rhythm? Well, the answer is yes. This was shown by Gary Ashton Jones back in 2001, where in anesthetized animals, he showed
that these noradrenergic cells were more active during the dark phase. Mind you, these animals are nocturnal, so this is their active phase. These showed that they were more active. So this was just looking at spontaneous activity, and while this is an extremely foundational piece of work, there's some questions that could be answered with today's technology available. So this was kind of the first goal of my dissertation project, was to look at this. So the goals in this project was first to replicate this finding that the noradrenergic cells are more active during the dark phase. I also wanted to test the hypothesis that these noradrenergic cells were more bursty, thinking about that bursting activity during the dark phase. And then at this time, this is when my lab had just relocated from UAD to MUSC. And so we were setting up the equipment. We were just setting up the equipment, and I was in my second year. I didn't really know how to do anything, so I really needed to optimize this methodology in preparation for the next project, which is what I was really excited about. So in our preparation for this project, we were calling it a circadian study, but the more that we learned, we saw that circadian really meant if we were looking at the full 24 hours, and we were only planning on doing the light cycle versus the dark cycle, and we learned that the correct verbiage for this was diurnal. So this is why we ended up going with calling it a diurnal project. So my very simple design for this project was I was using middle-aged wild-type Fisher rats. This is your classic albino lab rat. I had two rooms. My dark phase animals were on a reverse light cycle, meaning that their lights were off during the day. Again, these animals are nocturnal, so this was their active time point. And then my light phase animals, they were on a normal light cycle, so their lights were on during the day, and this is their inactive period. So the prep that I used for this was in vivo electrophysiology under anesthesia. For this, my animals were health-based in stereotax, and they were under isoflurane. Now, like I mentioned before, the locus coeruleus in human, it's very small. It's even smaller in a rat. It's rather difficult to hit, but I got pretty good at it. And for these recordings, I used 16-channel recording electrodes. I could pick up so many neurons at one time. It was really beautiful to watch, and I had a lot of fun doing it. So once I was confident that I had placed my recording electrode into the locus coeruleus, I began my recording protocol. This started with 15 minutes of baseline firing. The electrode is in the LC, and I'm just picking up spontaneous activity. After this, I hooked up the rat's foot to a stimulating box and elicited this very specific foot shock protocol. I borrowed this from the literature. Like I said earlier, the locus coeruleus, kind of the seat of arousal. I'm trying to arouse these neurons with this shock. And I was under the impression, based on the literature, that this increased the activity of these neurons. After this, this went on for 5 minutes. The shock was delivered every 10 seconds for 5 minutes. And then after this, I gave an injection of clonidine. Clonidine is an adrenergic receptor type 2 agonist. These neurons, these receptors are all over noradrenergic cells of the locus coeruleus. These receptors are GPCRs, and they are GI coupled, meaning that it inhibits the activity of these neurons. So I injected this. It takes a little bit of time to wash in. I gave it 25 minutes, and then this last 5-minute period was when I assessed the effectiveness of the clonidine to inhibit my neurons. Now, my criteria for clonidine inhibition was that this clonidine effect assessment period must be less than half of the unit's baseline firing. So just a little example for this, say that we have 2 neurons. We can see that this blue neuron, by the time that the clonidine effect assessment period comes, this neuron is clearly inhibited from the clonidine, where the red cell is none. So from this, I can confidently say that this blue unit and units like it are sensitive to clonidine, confirmed noradrenergic cells of the locus coeruleus, whereas the red cell, I can only say that it is not sensitive to the clonidine. Presumably, it might be one of those peristerulier cells that are reaching into the nucleus. So in this project, I recorded a total of 461 units. 70% of them were sensitive to clonidine, the remaining 30%. I did not include in this analysis, so I'm not going to talk about them today. So after I had finished my recordings, the electrode is down in the locus coeruleus. I gave it a little bit of a wiggle to increase tissue damage, and then I removed it from the brain. And then extracted the brain, cut it up, and sliced it and stained it with neutral redness stained cell bodies, and you can see the locus coeruleus really well. And then if you squint really hard or follow this blue line, my electrode track is right next to it. And so you can see it very faint, but then this tissue damage, that's from me wiggling it around. So by doing this for all of my animals, I can see exactly where in the brain I was recording. These images are from a rat stereotax atlas, and then these numbers indicate the location posterior to retina. So what's neat about this is I could confidently confirm that I was recording in the locus coeruleus, however, I was not recording in the same region. So with this, the first thing that I found was that these neurons are more active. There's more neuronal firing during the dark phase. So the first thing that I did is I just averaged all of my units firing over the entire experiment for both groups. One, so that I could have a general idea of what was going on, but also so that I could see that both groups were equally inhibited by the quantity. So what I was really interested in was looking at the baseline firing and the foot shock firing. So starting with the baseline, when I averaged all of the units' activity together, what I saw was that my units from the dark phase had significantly higher firing rate compared to the units in the light phase, and that my units in the dark phase had significantly lower interspike interval than the light phase animals. So, okay, this is great, checks out with the literature. Now, looking at the foot shock period, I saw a very similar trend. I saw that my units from the dark had higher firing rates, and they also had lower interspike interval compared to the light phase animals. Okay, this is great, these novel neurons are being aroused. But what I was really confused about was I was doing this foot shock protocol to kind of stress these cells out. I thought that this was going to increase the firing rate, and I was not seeing a significant difference between my baseline and my foot shock condition. So it was really humbling. Lily had sent me this paper where they were doing in vivo recordings in the locus coeruleus. They were stimulating the LC by way of the perforant path in the dentary gyrus. And they saw that a large percentage of them became inhibited by this stimulation. And Lori sent it to me and he said, hey, do you think that this could be something like what you're seeing? And I said, maybe. And so based on this, we decided that we needed to look at all of the units individually to see what was going on. Now, when we did that, we saw a ton of heterogeneous activity. So let me walk you through this heat map. So here on the y-axis, I have both of my groups broken down by animal. Each row is going to be a unique unit, a single neuron. Now on my x-axis, I have time. These are broken down into 0.5 millisecond bins, little squares. And the color of that square is going to indicate how many spikes are happening. So the warmer colors means more firing, cooler means less. So I think the first thing that we can appreciate by looking at this is just the heterogeneity that we see in the activity, but specifically looking at the foot shock period. So I'll remind you, the data that I was showing on the last slide, these are averages from all of the units within my group. But it's interesting because we see that some units are increasing their firing and some units are decreasing their firing. And so what we decided from this was that by averaging all of these firing rates, we're masking any heterogeneity that comes during this foot shock response. So because of this, I decided to split up my units based on their firing rate in response to the foot shock. And when I did that, we saw heterogeneous responses. It looks a little something like this. So on my x-axis, I have my light and my dark groups. Each of these dots represents a unique unit. On the y-axis is going to be % change in firing rate from baseline. All of the units down here are going to be the ones that decrease their firing rate in response to the foot shock. The ones up here are the ones that increase their firing in response to the foot shock. So this was really interesting, and we could say, wow, there are two populations of these noradrenergic neurons that are having physiologically distinct responses to this foot shock. How interesting. So what we were curious about was, could we predict what the response to the foot shock was going to be based on their firing during the baseline period? So what I did to do this is I essentially broke these up into 4 separate groups. My dark increase, dark decrease, light increase, light decrease. And then by averaging their firing rates across all of the conditions, we saw some pretty interesting things. So during the foot shock period, you can see that within my light phase groups, there are significant differences during the foot shock. However, during the baseline period, there was no significant differences in this. So from this, I concluded that the baseline firing is not indicative of how these units are going to respond during the foot shock. Okay, so everything that I've been talking about up until this point has been looking at single unit activity, saying this is the unit and this is how it behaves in each condition. Now, while this is great, we get a lot more richness of the data by looking at these bursting events. I'll remind you that a burst is a lot of spikes and not a lot of time.
This is needed to increase noradrenaline release to downstream targets in the brain. And this is so that an organism can mount an appropriate response to a stressful stimuli. So within looking at these bursting characteristics, there's a couple different properties that I looked at. A set of them is kind of looking at all of the bursts that are happening. These are things like burst count, burst rate, interburst interval. This is going to be how much time is in between the bursting events. And then there's also properties that I could look at within a single bursting event. These are going to be firing rate, interspike interval, burst duration, and spikes per burst. So for this project, I had a total of 2 groups and 2 conditions, and I was looking at 7 different characteristics for a total of 28 comparisons. I'm not going to bore you with some shifts and stats, but I do want to break it down for you, the relevance of it. And I'm going to use this term bursty. This is not a technical term, but I think that it's a lot more intuitive thinking about all of the characteristics that go into this burstiness. And this has to do with certain things increasing, certain things decreasing to indicate more burstiness. So yeah, these are the terms that I'm going to use to describe what I saw. So what I'm going to do is I'm going to compare my dark to my light, and I'm going to do it between my 2 conditions, the baseline and the foot shock. And then I'll be using more bursty and less. So during baseline, what I saw was that my units during the dark phase, they were more bursty compared to the units during the light phase. And I think that we can kind of get that from the averages that I showed on the previous slide. Now during the foot shock, same trend. The units during the dark phase were still more bursty than the units during the light phase. The next thing that I did, well, okay, so then yeah, we know that they're more bursty during the dark phase, during baseline and foot shock. So the next thing that I did was I wanted to compare the shift within groups between baseline and foot shock. So in my dark phase units, I saw that there was no change in bursting properties. They were bursting one way during baseline. They were bursting the same way during the foot shock. So during my light units, there was something different. During the foot shock, they became slower and they became longer, which was very interesting. So from this, I concluded that during the light phase, there's a greater change in burst dynamics because we're seeing that there's this shift where in the dark phase, we're not seeing any significant differences. Okay, so to conclude what I learned from this first project, I successfully replicated some previous data showing that these noradrenergic neurons are more active during the dark phase than in the light phase during baseline and during foot shock. This is what I think was the most interesting, but there's this heterogeneous response to the foot shock stimulation. And now what I want to remind you is that I was not recording in the same region of the locus coeruleus, and thinking back to kind of the anatomy of it, I think that this could be reflecting possible region-specific noradrenaline demand to this particular stimulus. And then what I showed was that the noradrenergic units in the dark phase are more bursty. And I think that this kind of gets at that intrinsic diurnal circadian rhythm that exists in these cells, despite that the animals are anesthetized, that these neurons are primed and they're ready to burst, even though the animal is not active and depleting. And by the same token, I think that that's why we're seeing that decrease during the light phase, that their bursts become longer and slower because they're not in their active phase. And this could potentially be to prevent hyperexcitability. Yeah, they cannot have this heightened burst response. Okay, let me get a little sip of water real quick before we get into project 2. But yeah, so this was great. Really fun to do. I learned a ton because the locus coeruleus, it was really hard to hit, so I was able to get my skills up and really prepare me for the second project, which was the impact of ovarian hormone deprivation on locus coeruleus physiology in this Alzheimer's disease rat model. So starting with Alzheimer's disease, Alzheimer's is the 7th leading cause of death in the world, and in the United States alone, it impacts 6.5 million individuals. Now, we typically think of Alzheimer's impacting older individuals with the average age of diagnosis being 65. Now, Alzheimer's disease typically presents with symptoms like memory loss, general confusion, and lack of executive function. It can be really frustrating for someone that's dealing with this. Now, Alzheimer's disease is a progressive neurodegenerative disease, meaning that it gets worse with age. Now, again, because we typically think of older individuals having Alzheimer's disease, but what's shown in the literature is that the pathologic protein accumulation that's ultimately causing
these cognitive deficits is starting as early as the 40s. Now, the pathology that I really want you to know, so pathology is starting as early as the 40s. And while there aren't any severe cognitive impairments that are happening here, there is an increase in arousal-related symptoms like anxiety, irritability, and sleep disturbances. So when I say pathological protein accumulation, what I want you to know for this project is mainly hyperphosphorylated tau, or p-tau. Now, I want you to know about this one because this is the first protein that starts to accumulate in Alzheimer's disease. Now, you may be wondering, where is this p-tau first accumulating in Alzheimer's disease? Well, it's in the locus coeruleus, and this is happening as early as the 40s. So there's not a ton of literature looking specifically in the locus coeruleus, but the general idea is that p-tau disrupts locus coeruleus physiology based on the preclinical human data in Alzheimer's disease. It's hypothesized that the locus coeruleus is hyperactive, and this is what contributing to those arousal-related symptoms like anxiety, irritability, and sleep dysregulation. And then this is being confirmed by heightened levels of noradrenaline in the cerebral spinal fluid. Now, zooming in and looking at the basic science work in Alzheimer's disease models, the general consensus is that p-tau disrupts physiology. This has been primarily done in cortex and in hippocampus, but there is one study looking at the effect of p-tau in the locus coeruleus. This is in the LC of a rat model that shows p-tau in the LC. And what was found was that the transgenic animals, these are the guys that have Alzheimer's, was that in basal firing of the LC, that their firing is actually decreased, whereas in burst firing, it's increased. And so this suggests that there's a lot more nuance going on within these models than what is hypothesized to be happening in humans. So getting back to Alzheimer's disease, my grandmother had Alzheimer's disease, and a lot of grandmothers have Alzheimer's disease because it's about twice as prevalent in women as it is in men. And not only is Alzheimer's disease more prevalent in men, but it's also more severe. Women have been shown to have more neuronal loss, and they have more molecular pathology compared to men. Now, there are many things that are believed to contribute to this discrepancy, but the overwhelming majority believe that the consensus is that once a woman goes through menopause, that her risk for Alzheimer's disease significantly increases. And this has been shown in many Alzheimer's disease models in many regions of the brain, that ovarian hormone deprivation or loss that happens similar to in menopause increases the AD pathology, the physiology, and the cognitive impairments that are seen in these models. Now, when I talk about menopause, I'm actually talking about the menopausal transition. This can take on the order of 10 to 20 years. It's a transitional period. So if we think about it on the timeline of Alzheimer's disease, now, while the average age of menopause is 51, we know that it's a transition, and perimenopause is starting early in the 40s. Now, by the time that the woman goes through a menopause into later in life and she no longer produces the ovarian hormones, if the AD pathology is already on board, it's become so severe and the cognitive deficits are so apparent that any type of therapeutic intervention is not going to be as beneficial as it would be if it was done earlier on. So this is what is considered the critical window for treatment, this early time point of p-tau accumulation and the early onset of menopause to really, it's really got to have the most benefit before the cognitive symptoms and the molecular pathology get far advanced. I also want to note here that the locus coeruleus, there's a little bit of literature showing that estrogen, the primary ovarian hormone, is inhibitory in locus coeruleus. So I just kind of want you to hang on to this in your brain, because we're going to be talking about it a little bit later. So taking all of this into consideration brings me to the goal of this project, which was to test the hypothesis that menopause will exacerbate locus coeruleus dysfunction in Alzheimer's disease. Now, any good project is only as good as its models. So let's talk about modeling menopause and Alzheimer's disease in a rat. So my lab uses the transgenic Fisher 344A rat. This rat harbors mutations in the amyloid precursor protein of the Swedish flavor, and also an exon 9 of presenilin 1. Now, this rat has been around for a little over 10 years, and it shows a bunch of molecular, physiological, and cognitive deficits that are similar in humans. This is a very well-studied animal, and the model is very robust.
Now, specifically, what's important for my project, and really novel about this model, is that it endogenously expresses p-tau. And I really want to put this home that this is the only model available on the market that endogenously expresses phosphorylated tau. Now, you may be curious, where is this p-tau first accumulating in the brain in these rats? Well, it's in the locus coeruleus, similar to humans. So I want to note that this p-tau is accumulating as early as 6 months in these animals. And then not only is it showing this molecular accumulation, but it's also showing physiological disruption. This data that I was talking about earlier, these recordings in the locus coeruleus were done in this model. So I say all of this just to really hit home that this is the preferred model for studying the changes that happen in locus coeruleus physiology in response to p-tau accumulation. So next, studying menopause in a rat is a little funny because rats don't actually go through menopause, but this can be induced by mimicking the hormone deprivation that happens during menopause by surgically removing the ovaries. This is called an overoptimization or an OVX, as I will be referring to it as. So going back to the goal, to test the hypothesis that menopause will exacerbate locus coeruleus dysfunction in Alzheimer's disease, I'm sure you're thinking, all right, easy, take your fancy rat model and take out the ovaries, induce menopause. And yes, this is the experimental group. And so we know that in this model, that the locus coeruleus is accumulating p-tau as early as 6 months, and the LC has physiological dysfunction. And so thinking about that goal, to exacerbate this dysfunction that's already on board, what we did is my wonderful lab manager, Natika, she overactimized these animals at 3 months, and then we let them remain hormone deprived until they were 6 months old to ask the question, will this menopause make the AD that's already on board, will this make this worse? Now, this is not a novel concept. Like I said, this has been done in various AD models and in other brain regions, and it shows that it does indeed make the AD worse. Okay, so one group is great, but we need some control. So naturally, I'm going to have some wild type controls. I'm saying wild type, these are non-transgenic rates, but I'm going to say wild type for simplicity. So I have a group wild type that were also overactinized at 3 months, remaining hormone deprived until they were 6 months. And then I also had some ovary intact animals. And so these guys had their ovaries, they had circulating hormones. And because this project was focused on hormones, this was something that was very important to control for. So to do this, I did all of my experiments on these girls while they were in proestrus. It's a time of high estrogen. And I thought that this would just juxtapose the hormone deprivation group very well. So now, what was really important to do was to confirm that my overoptimization, that this was in fact depriving my animals of hormones. This can be done really easily by weighing the animal uterus after the experimentation. So what we can see here is that my proestrus girls, that's going to be noted by these P's, their uteri, they're big, they're juicy, they're full of those hormones. Whereas my overoptimized animals, their uterus are small, they're kind of shriveled up. They have been deprived of these hormones. So that was really nice. Checks out, okay. These animals are in fact hormone deprived. I also want to note that based on the last study that I did, we decided to do these recordings while the animals were in their dark phase, so that we could capture the most amount of locus coeruleus activity. Okay, so again, I did anesthetized in vivo electrophysiology recordings. My protocol for this was very similar. The only things that were different is my baseline firing was a little bit shorter, it was 10 minutes. And then I introduced this 10-minute post-shock rest period, as I'm calling it. And so this was, I introduced it to kind of give the neurons a chance to recover after this foot shock stimulation to see if their activity would potentiate or depress. But then I injected my clonidine, let it wash in, and then the last 5 minutes, I assessed the effectiveness of the clonidine. So for this project, I recorded a total of 853 units. 82% of them were sensitive to the clonidine, confirmed noradrenergic cells, and the remaining 18% I'm not going to include in today's discussion. So based on the last project, the first thing that I knew that I needed to do was look at all of the units individually to see what their activity was like. So this heat map is similar to the one that we were looking at before. The x-axis is actually going to be up here with the recording protocol timeline. And then the y-axis is similar, broken down into my groups. And each row is a unique unit, but instead of breaking it up by animal, what I did is I sorted them based on firing. So
highest firing units are gonna be at the top of the group. It just kind of makes it easier to visualize. So before I get into the analysis of it and kind of the numeric quantification, I think just looking at it, we can kind of appreciate some of the differences. So the first thing that I really noticed was that my overoptimized animals, there's a lot of red in this graph. And this indicates that there's a lot more high level firing. The second thing that I noticed was that my transgenic animals, these guys right there, there's a lot more blue. And so this indicates that there's a lot more low level firing. And so we'll kind of get back to the quantification of that, but just like looking at this, I think that we can draw that out first thing. So the first thing that I found was that hormone deprivation increases baseline firing. We're starting with this first condition during the baseline period, looking at firing rate. So here on my x-axis are my groups. All of these dots represent individual units, unique neurons, and then these bars are averages for each of my groups. So the first thing that I saw was that there was a significant main effect of hormone deprivation where my overoptimized animals, they have significantly firing rates compared to my ovary intact animals. Now next, yes, and so shows the hormone deprivation increases baseline firing. But the next thing that I looked at was the interspect interval. Now with this, I'm seeing a slightly different trend. Where similar to the firing rate, I'm still seeing a significant effect of hormone deprivation, where when the ovary intact animals, they have a higher interspike interval, meaning that their spikes are happening less frequently than my ovary overoptimized animals. But then I'm also seeing a significant main effect of genotype, where my transgenic AD animals have significantly lower interspike interval. And so this is indicating, that their spikes are happening at a higher frequency. Okay, so the next thing that I did look at the foot shock and the rest period. Now, if you can remember from the last project, I saw this heterogeneous response to that foot shock. And so I was kind of anticipating that when I went to look at this, and that is exactly what I found. Now, for the sake of time, I'm not going to go through all of these data. We'll kind of go through it when I talk about the burst, but I want you to take from this is that there are physiologically distinct populations of these noradrenergic neurons in response to this foot shock. Okay, so next I'm going to go through those bursting dynamics that we talked about earlier. So for this project, I had 4 groups, I had 3 conditions, and again, I was looking at 7 different characteristics. So this was for a total of 84 different comparisons, and so we're not going to go through all of this because I did take 3 weeks and it's long. So again, what I'm going to do is I'm going to describe this in terms of burstiness, again, just because I think that it's more intuitive to understand. So what I'm hoping that I can convince you of is that ovarian hormones potentiate bursts during the foot shock, and that my transgenic AD animals, they cannot potentiate bursts post-shock. So to do this, I'm going to walk through all 4 of my groups. Keep in mind this wild type pro-estrus group, this is really my control throughout all of my groups. And then I'm going to go through each condition, and you can kind of think of this as the baseline being the in-group control, the foot shock being the treatment, and then the post-shock rest being the post-treatment. So starting with my wild type pro-estrus during baseline, I'm going to call these bursting. They were doing the noradrenergic thing, and they were bursting. Then during the foot shock, what I saw was that these bursts became more bursty, faster firing rate. They became longer. These neurons are getting aroused by this foot shock. And then during the post-shock rest, they maintain this burstiness. It's remained significantly elevated compared to baseline, but was similar to what was happening during the foot shock. So next, looking at my transgenic pro-estrus animals, during baseline, I saw that their bursts were less bursty. They had less spikes, the spikes were happening less frequently. But during foot shock, they got it up. They became more bursty, faster firing rate than they were during baseline. But then during the post-shock period, they returned back to their baseline levels. They were not able to maintain these potentiating bursting properties like the wild type animals were. So in my wild type overoptimized animals, these guys, they were very bursty. I think that we could have appreciated that just from looking at the heat map that we saw. So they were more bursty compared to the pro-estrus animals. Then during the foot shock, their burstiness didn't really change. It was maintained. It was similar to how it was bursting during the baseline. But during the post-shock rest,
this is where they really picked it up. They had this type of potentiation during this post-shock rest, similar to what is seen in my wild type pro-estrus animals. Now, finally, in my transgenic overactimized animals, these guys were more bursty compared to my wild type pro-estrus. They were a little less bursty than their wild type overactimized animals. Now, this is where it gets weird. During the foot shock, their bursts became slower and they became longer during the foot shock, which was really interesting. And then during the post-shock rest, their bursts returned to the baseline. They did not continue bursting in the slow and long fashion, but they returned to their bursting levels like they were in baseline. And so, again, to kind of get at these 2 points that I'm trying to make here is that these ovarian hormones potentiate the bursts during the foot shock. And so I'm specifically talking about these two groups during this condition where both of their bursts increase in direct response to this foot shock. Whereas these overactimized animals, these guys, their bursts stayed similar to how they were during baseline, and these guys got slower. The second point that I'm making is that my transgenic animals, that they cannot potentiate their bursts after this foot shock. And so I'm talking about these groups where their burst levels returned back to what they were during baseline before the shock, and how this is different than my wild type animals, whose bursts became more bursty and more bursty compared to what they were during baseline. So to kind of conclude everything that I've talked about in this project, what I showed was that ovarian hormone deprivation increases baseline firing. We saw that just in the firing rate. And also saw the baseline firing frequency is heightened in these transgenic Alzheimer's disease animals. And we really got that from the interspect interval. This is showing more of the firing pattern of that. And then the last thing that I talked about here was that these ovarian hormones potentiate bursts during that foot shock. That's going to be with these pro-estrus animals right here. And that my transgenic animals, that these units cannot maintain their bursts post-shock. And so again, thinking about how these burst levels return back to how they were during baseline. Okay, so now I'm going to get into the philosophy of this PhD and talk about how these data relevantly add to the field. So breaking this down into the two projects, what I saw was that these more adrenergic neurons, that they show this higher firing during the dark period. And so remember, these animals are anesthetized. And so this really shows that this diurnal rhythm, that this is intrinsic to these neurons, that they will continue to tick at this rhythm, regardless if the animal is awake, behaving, anesthetized, and they will continue to have this increased firing than they would during their inactive period. And then the second thing is that, so thinking about those bursting patterns, seeing that during the light phase, that their bursts became slower and longer. And what I think is happening here is that because these animals are in their inactive period, this might be kind of a preventative mechanism to mitigate any type of overexcitation that happens when this animal is in its inactive phase. This could be detrimental for sending more noradrenaline than what's needed during the animal's inactive period. And so what I want you to take away from this project is that just thinking about time of day differences is extremely important, especially if you're studying any type of disease. Especially if you're studying any type of disease that has circadian dysregulation, which is most diseases. Now, thinking about project 2, what I hope that you can appreciate is that these ovarian hormones, like the estrogen, and thinking back to estrogen having this inhibitory role in the locus coeruleus, this is inhibiting this noradrenergic excitability. And what I'm thinking in the, I guess, context of menopause is that the lack of this inhibition could really be contributing to those arousal-related symptoms that we see, like anxiety, irritability in menopause. And then what I'm also hoping that you can see is that bursting during foot shock highlights a role in adaptive stress. Yeah, I think that was supposed to be a different bullet point, but I guess what we're seeing here is that this arousal response that's mounted during the foot shock, that this seems to be mediated by those ovarian hormones that are on, or no, there is some literature kind of supporting this. And so just again, underscoring that role of ovarian hormones in arousal responses. And then finally, the last thing that I want to talk about is how these transgenic animals do not seem to be able to maintain this arousal level post-shock. And I think that this might be suggesting insufficient noradrenaline release to the rest of the brain for stress recovery, thinking about in Alzheimer's disease with T cell accumulation in the locus coeruleus. And then the last thing that I want to talk about, which I think was the most interesting, was this heterogeneous response that I saw in response to this foot shock. And I kind of touched on it earlier with the conclusion, but I think that this could be indicating kind of subpopulations in the locus coeruleus. I mean, it would make sense that, say, I'm reporting from one cell that might project to the amygdala, and maybe it does not need increased noradrenaline in response to this foot shock, where maybe the cerebellum would in that case. So I just thought that that was really interesting. And there's a lot more work coming out to support this heterogeneity within the LC. And so with that, I want to thank my amazing mentor for all of her encouragement and support over the last few years. I want to thank my committee for their guidance, especially during this year. I want to thank my lab. I want to thank the department for all of their emotional support. I want to thank you guys all for listening. I want to thank my parents for their support through my entire life. My friends that have really been in it with me through thick and thin. I know what's going on. My boyfriend for his joy and love. And everyone for coming here today. Thank you so much.
Really great talk. It's awesome to see it all come together. I was wondering, you said that you think in your kind of conclusions there for project two, some of the changes in the transgenic animals might underlie some changes in activity. Are there behavioral studies that use those animals that have shown that some kind of those behavioral deficits we anticipated occurring are also present?
Yeah. So the thing with this model is that those behavioral cognitive impairments are starting pretty early on. Anxiety, they have attention. We see that in later on for the profession and so that was related to pretty top really.
And then just kind of a follow-up for project one, I was just wondering, have you or anyone else done like separation and really not that back to the fire property?
Yeah, I haven't personally. That would be something that would be really interesting. I would imagine that would influence the circadian rhythms as well. I know that people have done more longitudinal studies over it, and then during a long active period, that people don't ask about it. And so I'm curious how you be able to help with that. I've never done the sleep deprivation. But yeah, it would be really interesting. Maybe you do have Yeah. I do know that there are some studies that they have done. I guess they have done that where it's like dark dark. And it does suggest
Great job. Yeah, that was amazing, and I'm very proud of you. But two kind of questions. One, I got at the end. Has there been studies of the neurons in the LC, like just any imaging of the LC neurons? Yeah, in relation to like sleep.
in relation to sleep.
Wake, Alzheimer's.
So imaging, are you talking about like accumulation? Are you talking about
Like calcium imaging, like sick, like activity.
Man, I don't know off the top of my head.
It seems like it's very hard.
Yeah, yeah, it is. That would be super interesting. That would be a really good way on exactly what happened.
Yeah, you can separate out those.
Yeah, there's not a whole lot of work. I guess like there's more work now looking at the heterogeneity of it. There's one graduate student in Jersey that she's like breaking down things like like that. I'm super excited about her.
Yeah, that was great. Also follow up. When you were talking about earlier, we had that heterogeneity and seeing that and reporting from different sites. Yeah, I was curious, had you tried to combine or look at, I guess, separate the data based on sites that were similar? Yeah, like the amount of data analysis you did.
Yeah. So that's the thing is that I'm actually love that to with the diurnal stuff. So it worked out interesting because for the dark cycle animals, I was really recording in a similar spot for those.
compared to the light cycle animals. And so that could definitely be influencing it. Yeah, yeah.
We had a smaller percentage of,
Units that increase.
yeah,
Yeah, yeah.
it was like a massive, that's why I was curious, it was kind of like a 30, whatever, and then there were 70,
I know.
13.
I know. And so like thinking about this, so I guess like, separate these guys, like with these in here, I'm really more in the top for DLC, the dark cycle animals, and in the lower part of the light cycle. And so like, if I could control for this, I would love to, but like, I'm really happy every time that I hit DLC because it's so small. Now I guess like physiologically comparing them, I did see the prelim analysis and didn't see a whole lot. Compared with the diagram, with the menopause project, I have a lot more, detail for that. Yeah, that's great, yeah.
Can I just follow up, because that was my exact question, also, great job. With the, like, heterogeneity, like, is there any work yet to suggest, the more, ventral parts project to the amygdala?
Yeah. There's one paper from, I think it's in Beach Boards, from 2015, and they did some retrovirus tracing. And so, I mean, it's one study, but yeah, they were showing that, this wing projects here, this wing projects here. I can't remember all of them because there's a lot. But yeah, so there's that, and then there's one other study kind of completing that. And so it's really interesting to think about, there's only, 1,500 cells in this really small structure, and then maybe it's like a cluster of like 300 is responsible for the amygdala, and like if I never get that report, I don't know what's going on. So yeah, there's like a whole world of opportunity with us.
Pivot question. You're talking about how like menopause is not like something that happens automatically, right? Like it happens over an extended period of time. But taking the ovaries out, is immediate, right? Like it takes that energy out. So is there any way like you kind of maybe like perfect world, you could do the experiment again where you could extend like slowly decrease that?
Really got that like chronic deprivation that happens. So I guess like just thinking about this that happens is, well, I guess taking the ovaries out does like initially there's that initial impact, but then it's like the rest of the body and the brain needs to adjust to that change that's happening. So there's going to be like the reduction of the hormone is going to stop directly, but the receptor landscape throughout the entire brain of the body is going to have to adjust to that. And that's going to take a little bit of time. So as far as like modeling that chronic deprivation, there is this model, it's BCB, it targets a follicular stimulating hormone and slowly kills it. There's some studies that are used on that. Well, I think that that's good to model the gradual deprivation that happens, but you have to keep in mind that like, this is introducing another drug. So what effect is that going to have, especially if it's targeting FSH? So then I know I feel like that's a whole other thing you have to control for people are doing it. But yeah, so it'd be cool. It would be super cool. Okay, so from the I appreciate it. But do this research. But I'm interested, okay, so when you see patients, especially all of them, they're coming in sometimes. I'm very interested in their quality. That I can't use them. It's no work. You can give it to ADHD, not give it to friends and people that are either of them. Others, it helps calm it down. So where does that play into effect in the fall? So the reason that I was doing it in my study, so this in the literature is pretty standard for targeting the LC. This is a really good way to like identify the arm fat reporting before activated cells in the literature. And so that was really the main goal, using the quantity to be able to chronologically say, yes, these are these reporting on. As far as using it as a medical treatment, I guess because it inhibits the neurogenetic cells of the ways, I guess that would probably contribute to mitigating those arousal responses. And I guess just thinking about it temporally, I would think taking that maybe in like the middle of the day so that it can have that decreased activation later in the day when those arousal symptoms are rising, that could be ethical.
I mean, I'm just thinking like, you do like,
and that's what,
the very small thing, one night. Yeah. And the other question was, as far as yeah, so I guess the big indicator for that, because like the LC is super small, so it's super hard to hit. There's a couple of different, I guess like physiological identifiers that I use when I'm looking for it, when I'm actually doing the reporting. One of them, and I didn't really talk about this at all, but one of them is the ventricle. When I go through that, I can hear it. And it has a pretty distinct sound, but then this region that's directly lateral to it is on the nucleus of the trigeminal nerve. So this nerve is right here. And when you're in it, if you have your left foot in it and you move the jaw, you get a really pronounced response. And so that's a good indication where I'm at relative to the LC, and then I can move my left foot into the LC. I can kind of look for that tonic firing, and then that phasic firing can be induced with a little toe pinch. And then I use that kind of like say, okay, I think I'm in the LC, and then I do the clonidine induction, and then post hoc do the analysis, say, were these cells inhibited by the clonidine? Okay, more confident, more adrenergic cells, and then I do my histology and say, yes, this is where my life goes. And so, like in total, there's like 5 different methods that I usually say, yes, these are the cells that I'm reporting.
Two quick questions.
Yeah.
Great job on this project.
Thank you.
Have you considered, instead of describing things as bursty, calculating burst firing index using burst duration, like interval?
Yeah.
Yeah, I think, just Google burst firing index.
Okay. Yeah.
And you show really clear diurnal firing patterns under anesthesia.
Yeah.
Do you think that anesthesia is contributing to that? And is there any evidence that those same patterns exist in vivo?
I mean, in a way, yeah. So there is some really good work on that. On and definitely. Now there's a couple different anesthesias that have been used recording from the outside. Things I could go to different the that for it is a lot easier, and you can probably look at better control than accessible. And so most of the activity is the something that you have to consider with that is that's not getting up to intrinsic by another because they animal having all of the environmental stimulus that's coming out of the experimenter, it's just like perceiving everything. And so that is really interesting. And when the animal is sleeping, those things are sleep stages. And there's a lot of work that's done without sleep before we regulate those sleep stages. And so that's one of the things we're excited about this project is that by anesthetizing the animal, we can kind of mitigate all of those other things that are sleep stages. And with the behavioral level that comes in really just look at that, what are you doing kind of in isolation, but in the but there's so many benefits to do it.
It's just a whole bunch of congratulations, great job. There was one question, but it was a duplicate.
All right, thank you. I'm going to excuse all of you so we can move into the private space with Rosie. And I'm guessing maybe at 5:15, 5:30, we'll have some more questions.
Anybody hear me now? Anyone on Zoom hear me?
I don't know. Let's see. I'm sure that I have a. Yeah, my favorite one. Wait, hold on. Yeah. Oh, wait, let me put it up.
Yes.
I literally like this smile. Even more like.
No. Wait, oh my goodness.
I was in.
Oh, you're so better, dude.
Yeah.
Yeah, great for us.
Okay, I'm gonna, send you guys out. That's been a short exam.
Nicest soul on the planet.
Family, they had problems. Steve, are you still there?
Yes. And everybody else now.
Okay.
Ceiling mics on so that,
Can you hear me now?
I can.
And if you can figure out how to get, we couldn't hear Steve earlier.
I think I
Can you hear me?
Just
You should be able to hear me now.
Trying to talk, but
Give me
Not
just a
right.
few moments.
we'll share it.
Well, thank you.
System.
I lecture in this room a lot. Okay, so first program, Mike. Hey, can you give us a hello, Dr. Shea?
Hello, can you hear me?
That we can. Okay, and then our next
Okay.
one is ceiling mics. Jim, you want to say hi just over there?
Hello, can you hear me?
Can you, let's try one more time.
Hello? Steve, can you hear me?
Okay, so what that means is, let's try this. Try one more time, Jim.
Steve might have-
Hey, Steve,
might-
can you hear
might
me?
be frozen.
Oh, is Steve frozen? That might explain.
I can hear him.
Okay, perfect,
Okay.
okay.
Okay, we'll just leave it in this configuration.
Giving a talk.
I didn't realize you were asking me. I thought you were asking the people in the room.
Oh, good, so you
Yeah,
could hear
yeah,
that
yeah.
whole.
Yeah, I could hear that whole thing. I thought he was asking the people in the room if they can hear him.
Great, that means we are fully
They haven't
connected.
given her a formal offer. Second.
I'm so happy.
Oh, wait, UAB for Beth?
That's so good. What
That's
department?
Neuroscience.
Yeah, neurobiology, yeah.
Or about. I'm actually speaking at VCU next week, Saturday.
Oh, you are? On a Saturday?
Yeah, it's like the SFN chapter meeting.
Yeah. Well, Richmond's an hour or so from me. I'm in
Oh.
Charlottesville. Yeah. Okay, how do we do this?
So usually we start with outside members. So Steve, if you've got questions, you can fire away.
Okay, are we gonna be looking at this? So we still have the screen up so she can show us data?
Yes.
Okay, all right. So I have to say I was really impressed with the talk, and the dissertation is very well written.
Oh, thank you.
I don't have many comments. I thought it was great. Two things about the introduction though, or really even throughout the document, this is a stylistic thing, but there's 4 words that I hate to read in scientific writing, and I try to never write them myself if at all possible. It has been shown
Yes,
Mhm.
yes.
that you use that a lot, and it's not like it should be banned, but it should be used very sparingly because it's empty language, you know? It just clutters up your sentences without saying anything. You can just say locus coeruleus is a new associated with arousal levels or something. You don't have to say it's been shown that. And also, I never said this before, but I think the introduction is too long.
Okay.
It was longer.
It was.
Yeah.
Is there anything specifically that you think could be omitted from it that doesn't add any relevance to my project?
I think probably the stuff about signaling pathways for both estrogen receptors and especially the noradrenaline receptors. I think that gets in a little too much detail. It's a little overwhelming to take in, and then you forget what you read earlier. It's a dissertation thesis, so I think it's your privilege to stretch out here. So I don't really think you need to make any draconian edits. I just couldn't let it go unmentioned that it's kind of exactly what we warned you about.
Yeah, yeah, and I was thinking that leading into when I was gonna submit it, and I figured that it would be easier to take things out during the revision process than kind of try to add it back in. But yeah, I can most definitely take that out.
What is there is very scholarly and very well researched and cited. So I wouldn't. I don't really feel the need to, in order to graduate, that you need to edit that down. But you may find a few passages that you can squeeze out, you know. But it's not an absolute requirement for me because what you did was all really solid.
Thank you.
Um, questions. So okay, I have a question and an interpretation.
Yeah.
With regard to the increase in LC firing during dark phase.
Yes, yes.
Um,
With the single unit activity or the bursting?
the single unit activity. Either one really. That's kind of paradoxical in a way. But do you think it's because the animal's more stressed, or do you think it may be that it's trying harder to stay awake?
So you're talking about because the animal is anesthetized, that they're trying to stay awake. Yeah, that could definitely be influencing it. And I guess thinking about the recordings, I didn't notice obviously any differences in the animals when I was putting them down, like inducing the anesthesia. That would have been something that I should have paid attention to. Well, and I guess I have all of the data for what their anesthetic depth was and the time that they were under. So that could definitely be influencing what the activity is. But I guess thinking about the literature that's been shown in awake and behaving animals during the dark and the light phase, and then also in the anesthetized animals, is that this finding is still seen that these units are more active during the dark phase because it is the animal's active period. But yeah, the isoflurane could definitely be impacting it, whereas the units the animals in their light phase, their inactive period, maybe they would be sleeping, so they're like, all right, we're still sleeping, as opposed to the active period where the animals would maybe be really fighting that anesthesia.
Yeah, that's just what I wanted to point out, that you can interpret that in multiple ways, depending if you view it as a stressful related signal or an arousal levels.
Mhm.
Uh, so there may be some intrinsic clock either in LC neurons or residing somewhere else that tells them it's daytime, you should be up,
Yeah.
or it's nighttime. So it's their subjective active phase.
And it's probably a combination of both of those things, the intrinsic regulation of them, but then also the effect of like either giving in or fighting the anesthesia.
Yeah. A few data items I had. So can you go to the slide where you compare burst? I don't remember exactly what it was, but you showed firing rate on is in the second half of the talk. You showed firing rate changes for the four groups.
This one?
No.
In the
Transgenic
Oh,
for a
okay.
rat.
Yeah, the second part of the talk.
Oh, right here.
Yeah. I'm a little bit concerned about this result because it doesn't really add up mathematically, because ISI is the inverse of firing rate.
Yeah.
I worry that this might be exaggerated by I want to know what test you used here, and because the measure of ISI is a bounded measure that has a long tail.
Yeah.
And so it may be the statistic of the underlying distribution of the data might lead to this discrepancy in results.
The underlying distribution of the
Look
data.
at your dots.
Yes.
See how they're all clustered? There's
These are things that we've talked about
Yeah.
before,
Uh-huh.
right?
That they're all here, and you say that, how is that influencing this result?
So if you use parametric statistics, it will make assumptions about the underlying distribution that don't hold for that the data. But well, I guess what I'm trying to reconcile is the fact that you don't see a difference. You're not able to detect a difference between the TGP group and the TGO group. No, actually, the difference between the WTP and the TGP. Yeah, so there's a small difference in in the the firing rate that I guess is not significant, but the fact that you're essentially doing a log transform on the data um, from when convert to ISI, and it may be exaggerating that difference.
You're talking about this, you think that this comparison in the ISI is exaggerating the difference that's not here in firing rate? Is that what you're saying?
Yeah, it seems that these things should go together.
Yeah,
They should go together.
so, okay, so I spent a lot of time thinking about this, because there was a couple different ways that I calculated firing rate. So initially, when I calculated the ISI, this was like, it was pretty neat because it's like, okay, this agrees with the hypothesis to a T, but where I was calculating the firing rate, I was not seeing this trend. And so because of this, originally, so I ended up calculating firing rate in like 3 different ways. I know, Jen, we talked about this quite a bit. Me and Lori talked about it. I talked about it with Steve. I talked about it with Michael Kelberman. I talked about it with anyone that would talk about it with me because I kept getting this disagreement between my firing rate and ISI. So what I ultimately did with this data is I just took total spikes over total time, hertz firing rate. That's what it is. The other calculations that I did is I took the inverse of interspike interval. So for each unit, I had their average interspike interval. I then took the inverse of that, so firing rate, because they're the ISI is the inverse of firing rate. So then I had the firing rate for that unit, graph that, still didn't agree with ISI. So I said, okay, what else? Maybe I'll take the instantaneous firing rate. So I took every single ISI, got the inverse of that, and then averaged those together to get firing rate. Still didn't agree with this. And so what we decided from looking at this data is that with the firing rate, so thinking that it spikes over time, what I'm concluding from this is that because I'm not seeing a difference in the transgenic animals, that they're still, during the baseline, during this 10-minute baseline, they're still producing the same amount of spikes. But what the ISI is showing is that the frequency that those spikes are happening with, that's where the nuanced shift is. They're still producing all of the spikes in the given amount of time, but the frequency that they're happening with is a lot faster. And that's kind of what I concluded from that. But I ran the numbers on these so many different ways. If you guys have any suggestions that might be able to help with that, I'm all ears.
It may just be a matter of statistical detectability, but sort of statistical power.
You were talking about the non-parametric test. So what would you recommend would be the best Kruskal-Wallis?
If you were running an ANOVA, right? Is it Kruskal-Wallis, the non-parametric ANOVA?
Or Man-Whitney, like I have no idea.
Man-Whitney U is the.
Kruskal-Wallis.
Yeah, it's Kruskal-Wallis.
For one-way ANOVA, non-parametric.
Okay.
That's what I would use for that, for data that are distributed like that, where they're clustered at the bottom. So
Did you run a 1 ANOVA on this or a 2x2? Because this is really a 2x2.
Yeah, it was a 2x2.
Yeah, I've never run a Kruskal-Wallis 2-way, but yeah, that's probably how you want to do it.
Is the ISI calculated as mean?
With this one, yeah. So on the bars, these are the averages within the group, and then for each single dot, it's the mean for that unit. So each dot is a unit, all of the ISIs during the period.
So for each unit, have you tried calculating it as a median?
Yes, I did do that.
And did that look more similar to the inverse of firing rate?
So it didn't, but I also calculated the medians for the firing rate, which I think it was the instantaneous firing rate, and then graphing those medians. That looked like the ISI, but then I don't know, at that point I was so deep in calculations and I'm like, I feel like I'm messing with this data too much that I don't.
So can I ask how you calculated ISI? Because one thing about your data is that it's like looking at, you know, all of the variety of activity you've got across all of these units. You've got some animals that like don't fire at all, and then suddenly they start firing during the baseline, and the ISI is gonna be calculated from first fire to last fire, right? Not over the entire time.
No, so I mean, the ISI is the time in between every single spike that that unit is
But think
firing.
about your firing rate. If you're calculating firing rate as number of spikes per 10-minute baseline, but your ISI doesn't start till 5 minutes in. That's like when the first spike happens, and then there's a delay in the second spike,
Yes,
and
yes,
then a
yes.
delay. And so like it might not be perfectly inverse because the time constant isn't the same between the two
Yeah.
things. I don't know if that
No, that's definitely the
tracks, but
And because I haven't, I guess, like looked at the single unit activity of every single neuron in the menopause data quite yet, just because there's so much of it. But I know that in the diurnal, there were some units that they would be off for the first like 5 to 7 minutes, and then they turn on.
Right.
And so yeah, that makes sense. That the firing rate would be significantly shifted because it could have 2 minutes where the unit's not firing at all.
but you're still dividing over 10
Yeah.
minutes. Yeah.
So I
Which
guess
is why I hate doing it this way.
Yeah.
Because you miss all that richness in the data,
So-
right? Because you're squeezing all that
Yeah.
pattern
yeah.
Mhm.
into a single number.
Right.
And I think that single number is meaningless.
Well,
Yeah,
So-
here,
One way you
I don't
could
like
do
it.
is spin the firing
Yeah.
rate
We
data
talked about that. Did you
into seconds and
Yes.
then take a median of that.
So-
Then it should be the inverse
Bin
of
the
bias.
firing rate, so total spikes over time, within a single bin. And then look at it that way.
Yeah, so basically like make your calculation for firing rate, 60 times in a minute,
Okay.
and then average that across a session. And if you're getting, big lapses in activity, that should the data should then look more similar to the inverse of ISI, I think.
But it's,
looking at the big picture and beyond this technical issue, you have a very interesting hypothesis, which is that the spikes rearrange themselves, the same number of spikes rearrange themselves into more bursts and less regular firing. And that's a really interesting hypothesis that you can test more directly than this. That's kind of an explanation, a hand-wavy explanation for these data, but you can actually test that by comparing the variance in the ISI. So for each spike, you want to calculate the actually probably the coefficient of variance with CV, which is the standard deviation over the mean. You compute that for each neuron, and then compare that.
With the ISI, take the standard deviation of.
Yeah. Standard deviation over the mean of the ISI.
Okay, and then
It's
what
a
is,
commonly used neurophysiological measure called coefficient of variance. It's not just it's a general statistical entity, but it's often used to describe neurophysiological recordings.
yeah, that sounds like
And-
an awesome way to do
and
it.
yes, the calculating the mean is never gonna tell you
Yeah,
anything.
Yeah, well-
Because it's about the pattern,
yeah,
right?
Mhm.
So
yeah, yeah,
I think this is a good idea.
yeah.
Yeah, it just doesn't.
Yeah.
To me, when I see this, it doesn't mean anything to me.
Mhm.
Because when I look at your heat maps.
Yeah.
You can see in the heat map,
Yeah.
there's all kinds
There's
of
so
pattern
much
in there.
different
So you lose the pattern when you make a mean.
Yeah.
And the mean
I
doesn't.
totally agree, Lori.
Yep, thanks, Steve.
Yeah, so that's such a good idea. Yeah, because I was really just wrestling with the firing rate because I had no idea what was going on. And then by the time that I did get the instantaneous firing rate to look like the ISI, I just felt like I was manipulating the data too much because I'm showing a median, but the rest of the data is in means. And yeah, it's just because.
Well, you weren't manipulating, you were just analyzing it different ways, right?
Yeah.
And then you have to,
Yeah.
any scientist can analyze their data how they want. You just have to say why,
Mhm.
how you did it,
Mhm.
how you did the stats,
Mhm.
and the rationale for why you analyzed it this way.
Mhm.
That's why when you look in the literature,
Mhm.
people, especially for these kind of data, they're analyzing it different.
Yeah.
And when we looked at Michael's paper, his ISI did not match the frequency.
Didn't.
And I was saying to you, this
Yeah.
makes no sense.
Yeah.
Right?
Yeah.
And that's because it's a mean,
Mhm.
and the neuron isn't even
Yeah.
having that activity of that mean number.
Yeah. So I guess going back to binning the firing rates over time, and so that's just gonna be having a bin set, taking total spikes during that time. But you said averaging them between the units, but wouldn't that still kind of get at the same thing with averaging the ISI essentially? I guess I'm confused.
Yeah, so, so the problem with taking a mean, or sorry, you're not taking a mean of firing rate, you're taking total number of spikes over time, right? So let's say you get 1,000 spikes at, the last second, and 0
Right.
spikes otherwise,
Mhm.
over 1,000 seconds, then
Yeah.
your firing rate is 1.
Mhm.
But if you average in bins,
Mhm.
and then take the median of that, then it's actually it's probably not a perfect analysis.
I don't
Or even
think it
close to
is, right? Yeah.
I think that Steve's idea of looking at coefficients of variation is probably something that is worth doing.
Yeah.
Yes.
Um,
Or even putting it in a histogram.
Just about to say
Right?
histogram,
Put it
yeah.
in a histogram.
Yeah,
to
so
see.
put what in histogram? ISI or spikes
Both,
per time?
any.
Okay.
And
Just within each of your,
Yeah.
your base, your
Yeah.
flight, your
And
rest.
you decide what the duration of the bin is.
Yeah.
Right?
Yeah.
So, we're gonna think deeply about all this
Mhm.
when we are
Writing
working
the
to
paper.
publish this.
Yeah, and
Right?
something that I just want to like touch on with that, I think that bin analysis, but more so like a histogram breaking it up by time is something that you can't really appreciate in the way that I have the data split up. But while I was doing these recordings, I kept seeing what I was calling these oscillations of activity, where all of the units might kind of increase their activity and then decrease their activity. But some of the units that I was recording at the same time, they would decrease. When other units are increasing, they would kind of have this like asynchronous oscillation. And I think that looking at them like temporally in BIMs or in a histogram, that's going to be really a really nice way to look at that. And I mean, like you can kind of see it here where like these units are all getting active, those units are all like that. But like when I have it broken up into animals, you can really see it. So yeah, I think that that's going to be an awesome analysis for that.
Yeah, I think a really important analysis to do, and this relates to Lori's point that she keeps making about losing the temporal structure. I think you want to quantify the coherence or correlation among units, like because it looks like it's a lot higher in the overectomized groups than the proestrus groups. That lumpiness you're talking about. The oscillation that structure seems to be much more prominent in the bottom two heat maps.
Yeah, you're so right. And I guess also thinking about that, because that's something that I guess like I didn't necessarily think about in here. But so when I started the recordings, it was once I had gotten into the LC. Now, once I had done the diurnal project and was at this point, I was pretty good at getting in as quick as I could, starting the recording, but also thinking about how long those animals had been under, and then thinking about, say that we're looking at the baseline firing time point, say that like this animal maybe had been down for like 30 minutes before, whereas this animal might be down for like 50 minutes before. And so I don't know if those oscillations, the lumpiness would line up exactly, but I think it would be interesting to take like the highest firing bin and kind of line those up all the way down to the lowest and see what that activity looks like.
Or talk to Nate Baker about having that as a modifying or as a, what's the word I'm looking for?
Covariate.
Covariate, where you're
Yeah,
trying to ask whether the time since the time under anesthesia changes, is modifying your response. Because I mean, again, to Steve's point, like you're looking at the arousal circuit under anesthesia, that's does make sense.
Yeah.
Can I go next? I have to use the restroom, so.
I'm gonna actually use it. Well,
So, I mean, you gave such a great presentation. I'm so impressed.
Yeah.
Really nice job. And I thought your dissertation read well. For the most, I mean, it was 300 pages, so I didn't read every word.
Yeah.
But
The last like 100 pages is code.
You read all the code?
actually,
Steve, did you read all the code?
No.
Okay.
so that's one thing I wanted to mention. I think 150 pages of code is a lot. I think if you're going to include it, there's some parts that aren't commented. So I think if you're going to include it, you should make sure that each subsection is commented
Mhm.
well.
I was actually
Most
gonna
were-
suggest that maybe it be put in a, like, just have an appendix with a link to your GitHub, because
Yeah.
I don't know how useful it will be to people
Yeah.
in this format in
Yeah.
a PDF online where you can't easily copy and paste things. So
Yeah.
I don't know if that's allowable. I
That's-
assume it is.
That's my
I
suggestion.
don't even know if she needs to include it
I
in
know.
there. It's
I
up
have-
to you
I have
guys.
all of the code on my GitHub, so
Yeah.
it's in a repository. I just wanted to include it because like I taught myself how to do all of the
Yeah.
coding. I
I
did all of
totally
it.
get why you put it in there,
Mhm.
The
but I think
advantage of-
for,
Sorry.
no, go ahead. I just
The
think that
advan-
for it, it might be better.
Yeah,
The
probably.
advantage of putting it on a repository
Mhm.
is that if there's ever an issue with it,
Yeah.
which 150 pages, first time coder, there's probably issues with it,
Mhm.
you know,
Mhm.
no offense.
Yeah.
Oh, yeah.
There's issues with my code too,
Yeah,
I
yeah,
guarantee
yeah.
it. You can change it on the repository
Yeah.
and not ever say a word about it.
Yeah.
So I like having a repository
Yeah.
that I can modify, I can
Uh-huh.
track the modifications for
Mhm.
people to see.
And this will be permanent forever and ever after the embargo is
Yeah.
lifted. So like, just
think-
remember.
That point is a really,
Yeah, that
It's
is
a
actually
really-
a good
yeah,
point.
because it's like that
Okay,
forever.
okay.
That's a good point.
Cool.
And it isn't useful to people. I mean, it makes you feel like, oh, this
Totally
is part of my dissertation, and I get that.
Yeah,
Very
I'm
good.
definitely like emotionally attached to it, but I can.
I agree.
But I think you
am
can
too
put
with
in
my
the
code.
appendix, I think you can put a link to the GitHub, and that way it's forever linkable to if someone's.
That's,
And people will Google, I Google things and get super excited when I see something that a data that I need, and then it's a dissertation and it's embargoed and I can't even get it.
yeah.
But, you know,
Mhm.
so.
Yeah.
I think it'll come up and people will link to it and it'll be
Mhm.
forever useful, but I think it's forever more useful than a Git.
Yeah,
than-
yeah, I get that. I can definitely do that.
When I first opened the file to print it, I was like, it's like 350 pages. And I was like, oh my God, it's longer than even my already too long dissertation. It's like double the size. And then I realized it was all code for the second half.
Yeah, I can take that out.
So one thing, and you might have mentioned this, but how you were defining, you were doing analyses within bursts, right?
Yep.
So how did you define if this is a burst or not in the first place?
Great question. So in the literature, this was defined by Anthony Grace back in 1983. And I just had like a little tiny thing, talking about it. But yeah, so what he called as a burst, and this has been used in other literature. But so like going through the spikes, and I had my code written up to do this, but so it did all of the calculations based on inter-spike interval. So the start of the burst happened when the ISI between spikes was less than 0.008 seconds. And then the way that I had the loop set up is like, okay, once it hits that ISI and within a burst, it's counting the spikes, it's counting the time in between the spikes, it's counting the burst. And then once it hits an ISI that exceeds 0.16 seconds, that is the end of the burst. And so then that burst is like stored as a burst event, and then it counts up all of the bursting events that happen with my unique units.
And so was Tony doing, I know he did a lot of PFC recordings, was he doing LC recordings for this?
Yeah, so the definition for this came from Locus Aurelius cells, and then he was also characterizing dopaminergic cells, and I think that was in the BTA.
And have you looked, because it's, and again, this is like something that's a little intangible, but like imagine a cell's firing at 5 hertz already.
Mhm.
It barely needs to change
Yeah,
to be quantified as a burst.
Yeah.
Whereas a cell that's not firing at all.
Yeah.
Would have to change a lot.
Mhm.
So I guess, I don't know, have you looked at a lot of individual units to see if this ISI analysis for bursts is really fitting most of your neurons?
yeah, well, so I don't think that it is. I think that this criteria for burst event, I think that this is really narrow. And I think that we had briefly talked about this in one of my committee meetings, but ideally, what I would like to do was to be able to say, it's like, look at the activity of a single unit and say, what is its highest level activity? Okay, and like classify that as bursting phasic activity for that unit, and then find the burst for that unit based on that criteria. So yeah, personally, I think that this is extremely narrow. It was developed back when, but this is what's been used in the literature, specifically in the Alzheimer's model that I'm using. And so I thought that it was really important to use this criteria so I can directly compare it to past work.
Sure, yeah.
I think I told you
carried, you know, for decades.
Yeah.
Yeah, and I'm not asking, I mean, I'm not asking you to like go back and change any analyses. I'm just was curious.
Yeah.
I think it's worth, you know,
Yeah.
doing that.
Yeah, and I mean, like, that's the thing is like, we could totally rewrite bursting definitions for these cells because like now I have like over 1,000 neurons. And so being able to say like, this is how these units are bursting, I think like that'd be cool. That would be really cool.
Change in firing rate, like, you know, percentage rather than,
Yeah.
something like
We
that.
can get an army of master students in data science
Yeah, that'd be
and
cool.
just say, see what you
Yeah,
can
have
find
fun,
in here.
have fun.
I've sort
Yeah,
of,
so Rosie, I told you several times, I think that I'm not quite comfortable with this analysis for several reasons. One is that it was based, at least in part, on dopamine neurons, which I think burst quite differently.
Mhm.
I'm not a fan of using fixed parameters that aren't derived from your own data. And the third reason is there's a report in the literature. I don't know if it's out in print yet, but it's on bioRxiv, the Nelson Toda's work. The LC's community, we all send them a bunch of data.
2024 came out last year.
Yeah.
Yeah.
And so they have quantitative parameters in there, but I think the most important thing to do if you're going to do anything with these bursts is you want to assess its performance at detecting bursts as you see them. So you can have
Within the
people score, expert scorers to check a subset of the data and make sure it's catching. You can get false positives and false negatives off that.
Sure, I did do that. And so I'm trying to think, it was before I went to the Association for Clinical and Translational Sciences. I had part of my code, and this was, I optimized this back when I was still doing the single unit recordings. So I was only getting like 7 units at a time, so I could go in there and look at it. But the way that, and I don't have that image easily accessible, but the way that I did it is I had it print out a roster plot of every single unit to look at the firing. And the spikes, they were black, but when the code recognized a bursting event, it made it a color. And then I had it overlaid with the firing rate. And so I could see when the firing rate increased, there would be like a conglomerate of spikes that indicated a burst. And so I'm pretty confident in this code that this recognizing as bursting events based on this criteria. But yeah, I completely agree with you that this criteria it's really narrow.
That makes me feel a lot better. I guess I forgot that you had hand checked that. So that makes me feel a lot better.
Yeah, because that code, that took me a while, and it was Zach Irwin. He really helped me get that fixed.
I remember us looking at that in a couple of lab meetings because I was worried that it was just making up stuff.
It was.
right?
Well, it was for a little bit. It was saying that there was like a burst that had 238
remember.
spikes. And it was going on for 4 minutes. And I'm like,
Yeah.
what? That's not right.
Yeah,
So
right.
yeah. So every single unit, and I was going and I was making sure that it was working. Yeah.
Yeah, I remember.
I have a naive question for you, which is, for these spikes, how well are you able to resolve the actual waveform of the action potential?
Yeah.
And based on that, can you tell me what is the property of the potential that is changing when you transition from,
Good question.
you know,
Yeah.
wait,
into
that
these
doesn't
bursts?
sound like a naive question.
Yeah.
No, that's a really good
It's
question.
not, it's
Yeah.
the neuroscience teacher question.
Yeah,
I
yeah,
can't
yeah.
help it.
Okay. So, so the waveform's not even something that I've looked at yet.
Okay.
We're gonna, so I have waveforms for every single spike that I recorded. And the way that I did my processing for this is initially, like, it goes through and the way that I have it is it recognizes the unique units based on my spike sorting that I did. It labels those based on like the animal, the channel, the unit that it came from. And the properties that I pull out are the time that the spike happened. I think that there's one, like some other ones about, but the rest of the information is about waveform characteristics. And so it's like area under the curve. It's like peak to valley, valley to peak, all of these different things. And then there's like a train of numbers that also comes out. Then someone showed me that if you take that single row, put it into Excel and you plot it, it's the waveform. I don't know how to do anything with that yet. But so what I did in the processing is once I went through and kind of like correctly labeled all of my cells, their units, what group they came from, who they came from. Once they were labeled, I then took all of that waveform information affiliated with that animal, with those units, and I threw it into a different file. And so it's all saved, it's all encoded, ready to go. But I just haven't done anything with it. And Steve, that paper that you were talking about with Nelson Toda, not the most recent one that he's the last author on, but his stuff from 2018, he does a really
Yeah.
nice job classifying waveform properties. And
Yeah.
I want to say that it's in that McKinney paper from 2023. Some of the waveform properties as far as the width of the action potential can give me insights into what type of cell it is, if it's a fusiform or if it's a multipolar cell within. And so like that's going to be a really cool analysis to do.
So just from like a fundamental properties of action potentials.
Yeah.
What might you hypothesize? What would you expect to see changed as a cell transitions from these more slower firing rates into a bursting rate?
Yeah. Well,
Good question.
I guess with this project, what I've been thinking a lot about is potassium channels. And so I think that it would have something to do with that about, I guess, like, I don't know, like shortening the repolarization period. And I don't know, getting that cell ready to fire again. And at least like in my menopause project, that was what I was seeing, especially with the changes in bursting. We were thinking that that was like, could be a potassium channel directly influenced by them. And so I'm thinking that that could be seen, especially with those like longer, slower bursts. So
I have another basic, never mind. Okay. So in your menopause project, you talk about the loss of ovarian hormones, and in the ovex, this is a very sharp change. What is the aromatase expression in pattern in the locus coeruleus?
So aromatase is responsible for converting, is it estrogen to testosterone?
Testosterone to estrogen.
To estrogen. That's such a good question. I don't think I've read any literature looking at that. And from my understanding, I don't believe that I don't think that there's any work showing that the LC produces estrogen the same way that the hippocampus does. But I mean, like it could. And I'm not sure. So I'm not quite sure what the expression of aromatase is. And so I guess like thinking about that, are you getting at if there's a loss of ovarian hormones that maybe there's going to be like an increase in estrogen production?
So in your dissertation, you talk a lot about, you identify a number of estrogen concentration dependent events. You talk about it in the gonadotropin releasing hormone system and in terms of the LH and FSH surge.
Yeah.
I think you talk about it also in terms of the affinity for the estrogen receptor. And so, you know, when we're thinking about this like slow decline in ovarian production, is there maintained production in cells with aromatase expression? Because there's not just the brain, there's other organ systems that have aromatase. And I also think that there are variants that are not just testosterone, but pregnanolone, which is a product of cholesterol, which most cells have the ability to do that step. It's just rate limiting. So like, the difference between depletion, decrease,
like a gradual
and so it's more just, and especially because you also note in your dissertation a lot of regional and wiring, and I wonder if capacity for local estrogen production. It's a really like just a
no, and 100%. And I was just kind of like thinking about that because like we know that estrogen is produced in other regions of the brain. I'm thinking about the LC being like deep in the hindbrain, really like evolutionarily conserved. I would believe that more, I guess, like conserved areas of the brain are probably capable of producing those hormones more than higher level structures. And that's just me like thinking about it. And so like, I'm sure that would happen.
I don't even need you to put it into the context of this. It was more just like an interest question of, given all of the different, you know, regional and functional cell type and projection differences within the LC. What else?
And maybe Lori, you could help me out with the literature because I know that there's literature in the hippocampus looking if you
I was
overoptimize.
just gonna ask you.
And I can't remember in the hippocampus if it goes up or goes down.
So CA1 pyramidal cells make plenty of 17 beta estradiol. Yet when you take out the ovaries, you lose spines. Right? So removing ovarian hormones causes change in hippocampus, even though CA1 cells can make 17 beta.
Like a feed forward.
Well, so the thinking is that the ovarian estradiol is not what's growing spines in hippocampus and enhancing LTP and learning. It's ovarian estradiol in the plasma, going to the hypothalamus and releasing GnRH into the hippocampus and triggering local estradiol synthesis. So it's still a 17 beta estradiol. It's just not what's pumped out of the ovaries. It's not making its way to hippocampus. And the same is true here, right? With the locus coeruleus. The simple removal of ovarian hormones is not, your heat map shows it.
Oh my gosh.
It's profound,
Which,
right? It's profound.
that was something I was really excited about because there is like, at least as far as my understanding, one paper showing that estrogen has this inhibitory role. They were doing it in slice in overoptimized animals, and then they washed on 17 beta estradiol, and they saw this really stark decrease in activity. In that paper, they also washed on progesterone, and they saw that it increased activity, so that showing that estrogen and progesterone kind of have this dichotomous role in the LC. And so I don't know, just like keeping that in mind, because this project is removing the ovary, so all of the hormones. But I guess thinking about estrogen as the primary ovarian hormone having the biggest influences. Yeah, so that data was just like really cool to see.
And so I think that's where your summary and your conclusions, in my mind, you and I've talked about this before, you're very myopic in the little minutia details without stepping back and really saying that you're finding and just in wild type between proestrus and ovax, that is just not known. That is just not
Line
known in the
33, I
literature.
think, was the one I wanted to go back to. It was like a.
And
have it
so
written down.
I think, and again, Rosie, you and I've talked about this a lot.
32, maybe
Being
32.
able to say what your data mean,
This one, yeah.
right? There was a lot of detail about this, but what does it mean?
Yeah.
For the animal, what does it mean?
Yeah.
Is this one you wanted to ask? Well, I'll let you
I
guys.
have this one
Yes.
on my list.
So so coming up with this like burstiness, man, I wish that I would have talked to Pat like 2 weeks ago. Because burst index, like,
It's, it's. out
hey,
in the
but now I know,
literature.
but now I know.
But I still like the cumulative distributions that you didn't talk about.
So
But
those,
we will in your paper.
well, so I have all of the cumulative distributions in my pocket. If you want to talk
But
about them.
this is for the purposes of my questions, and maybe theirs too, this is good enough that you guys can ask.
So can I pause? So with this was just my understanding what I thought was important. And so this was me trying to like deduce the subtle shifts in all of these properties. And because like, I don't know, with some of the ones that became more bursty, they didn't become bursty in all of them, but maybe like all of them except for one. And then it got kind of difficult with some things if like firing rate was increasing, but burst duration was decreasing, and trying to like deduce that to see how they fit together. So that was kind of difficult for me to do. And I felt like, wow, I feel like I'm almost like.
And I think for a seminar like this, that's fine.
Yeah.
But being able to talk about what this stuff means.
Yeah.
Because otherwise it's just like noise, kind of. So I don't know what your questions were.
Well, I had more like the high level 30,000 foot view type question to just get you feel free to pontificate about. So you talk about, this whole thing is kind of that, so women are more susceptible to Alzheimer's, right? And that, you start to see this accumulation of these phosphorylated tau proteins, early in the 40s, maybe when perimenopause is starting to hit. And so I guess my question is estrogen really protective, or is it actually maybe a risk factor? Because I'm just like thinking about this as a person outside of this field, and it's like men have less estrogen and they have less Alzheimer's, and women have more estrogen. And when we become more men-like and we have less estrogen, that's when it's sort of revealed. But is it the case that maybe estrogen is actually promoting and not protective of?
Yeah. So I guess, are you thinking molecularly, physiologically, both of them?
All of them, yeah.
So I guess like for me, what I think it is that estrogen is like, all of the literature shows that it is this like potent neuromodulator, and that it is beneficial in a sense. And so I don't think that it's that women have estrogen and it's this risk factor, but I think it's that we have it for majority of our adult life. And then once we hit this age, then it starts to dip off and we don't have it. So it's like kind of like it'd be like having your leg until you're 40 and then you lose it. Like walking is going to be a lot harder. So just kind of thinking about that. as far as with Alzheimer's disease, and even like outside of Alzheimer's disease, just healthy aging for women. And it's not even just in the brain, but as far as like the cardiovascular system, with our bones, osteoporosis risk increases after that. And so, yeah, I wouldn't say that estrogen by itself is or like a risk factor, but I think it's that loss of it, considering that women have it and then they don't. And then there's a lot of research showing that hormone replacement therapy is extremely beneficial for mitigating the symptoms that come with Alzheimer's disease, but then also like the cardiovascular, osteoporosis.
So then how do you also make sense of your, I guess let's just look at the top line, your wild type proestrus animals becoming more bursty when they have the most estrogen circulating in their system?
Mhm.
Because
Yeah.
shouldn't it, you said it's kind of inhibitory.
Yeah,
Right.
yeah. So that was something that I have spent a lot of time thinking about this. And in the literature, I don't know if you're familiar with Susan Wood at university. Yeah, and so Cora Smiley, her postdoc that I did she trained me on doing those recordings. But so some of her work is looking at CRF. And at least from that literature that I've read is that estrogen kind of seems to enhance this like stress response. And I guess that could be increasing like why anxiety, PTSD, depression is more apparent in women. And so I think that there is like, at least from my understanding of the literature, that estrogen potentiates this response to stress. And so like I don't necessarily know if it's a good thing. But that seems what is happening. And I guess we do see it in these data.
Why could it be a good thing? Let's just assume that it is.
Mhm.
Why could that transition be a good thing?
Yeah, so I guess like in the response to foot shock, I mean, just thinking, say that these animals were awake behaving out in the wild and they had heightened levels of estrogen. Oh my gosh, they were being chased by something. Their locus coeruleus is all right, we gotta go, we gotta run. And they can mount a better arousal response to this stimulus. Whereas an animal with lower estrogen, they're not gonna be able to have the same response acutely to that stimulus.
So you think it's like not necessarily the firing rate at any given point is important, but it's like the delta, the change in firing rate.
That's what I like with this whole slide was trying to get at, was like from baseline to foot shock to post shock, right? Kind of how that shift was happening. And this wasn't even something that I was really thinking about until like the past 2 weeks, when I started noticing the differences in the shifts, because some of them they increase during that post-shock rest period, whereas like my transgenic animals are not. And so just like seeing how they're shifting. Yeah.
You talk, sorry, go ahead.
Go ahead, Rachel.
I was gonna say, you mentioned CRF a bit in your dissertation. I'm wondering if you can you just remind me how CRF acts on locus coeruleus neurons?
Yeah, I should know this a little bit better, because like I think that this is like one of the underlying things, especially with the foot shock. But I know that CRF, like one of the main targets is the locus coeruleus, and that there is this study in Cora Smiley. She was looking at its connection with the central amygdala. And I'm pretty sure that what she saw was that there was a heightened response when there was high estrogen. And like, I could know this literature better, but that is what is coming out of my brain right now.
So they express the CRHR1 receptor or whatever.
I don't know if it's that specific one, but the LC is very susceptible to
Increases activity in response to CRF. Or
I think so. I think that it's increased.
And in part, what's around the locus coeruleus, that's where those CRH projections have their most potent impact.
Yeah, interesting.
So they are GABAergic.
Yeah.
They release CRF, which is acutely excitatory, but they are co-released with an inhibitory fast setting. And so they disinhibit the LC through the pericoeruleus.
Disinhibit. So they're taking off the inhibition from the pericoeruleus. Whoa. Yeah.
Is at least one definite mechanism. Whether or not CRF also then can activate locus coeruleus neurons.
directly is not clear. Okay.
Okay.
I don't know that for sure, but I do know it can disinhibit through that.
But those are all great future directions in my renewal ROI. Yes.
Yeah.
So can I go back to this? Is it okay if I ask questions?
Absolutely. Yes, you're part of the committee.
What's the role of norepinephrine in learning and memory?
Let's see. It is needed and necessary. I know that there were some studies using novel object recognition where they ablated the locus coeruleus and the animals were not able to perform.
Yes.
As well.
And what part of the brain is needed for novel object recognition,
The hippocampus?
right?
Mhm.
Okay, so I'm gonna help you pull
Oh.
this circuit together.
Oh, well, I
Right?
mean, because the LC is projecting to the dentate gyrus.
Yes,
And that, the dentate has the most dense innervations from the LC, the most dense noradrenergic intervention.
and that circuit is critical
Mhm.
for novel object recognition and not novel object placement.
Mhm.
And in order to do that, the animal has to be aroused,
Yeah.
right?
Mhm.
And what do you know about learning and memory at proestrus,
Ah, it's a lot better.
right?
Mhm.
So couldn't you
Ah,
imagine?
Yes.
So tell me what I'm thinking.
So you're thinking, so maybe instead of like this foot shock, but just kind of thinking this like as an arousal
Yes.
stimulus, height firing.
Yes,
In the LC, and then thinking about those projections to the dentate gyrus and the hippocampus, that is going to facilitate increased learning when it's
and
aroused.
learning is heightened.
Yeah.
Hippocampus dependent learning is heightened at
It
proestrus,
is, it is.
right? So you have to be,
Yeah.
thinking about this as
In
a
the circuit,
circuit,
yeah.
right?
Mhm.
So back to Jen's question about, oh, I thought estradiol should be inhibitory, and maybe it is.
Yeah.
So you can have that delta, right? You can have that
Yes.
arousal,
Yeah.
right? The signal to noise,
Yeah.
right? So maybe it's controlling the signal to noise so
Yeah.
that you can have this
Yeah.
response.
Uh-huh.
So you can drop
Yeah.
norepinephrine and hippocampus and you can heighten.
Mhm. Yeah.
Okay, so tell me what's going on in TG then,
And TGOX.
any of them.
Yeah, well, so I guess with the TG Pro Estra, so their burst levels are lower, which I think is interesting. And this is something that I guess I'm still trying to like wrestle with, thinking about the single unit activity where I was looking at those bar charts with each unique unit being like, all right, this is how it behaves in this condition. But then looking at the bursting mechanism is kind of, I guess, like within a single bursting event in that. And so like there are definitely some discrepancies with this because I guess like I wouldn't have predicted this based on the average spiking. So just seeing the lower, I guess, burstiness, that there's less spikes in here. And with this, I guess what I was thinking about this was that this kind of agrees with Michael's data in the, he was doing in vivo locus coeruleus recordings in this transgenic model, and in basal firing, he saw that firing was lower in the transgenic animals. And so I think that this might be contributing to that. But I think that it's still interesting and again, kind of like underscoring the role of ovarian hormones in this like stress response that we're seeing during the foot shock. And then as far as it turning back to baseline levels, not being able to maintain this, I think that this could be that and this is not gonna be a technical term, but like the neurons, they have p-tau on board. And so like they're trying their best during this foot shock, but post they're like, oh, I'm tired, I can't keep firing like this. And I guess like thinking about it as far as if the animal was awake and behaving, like maybe they wouldn't be able to show like increased learning or some increase the way that wild type
Or it's
animal would be.
perhaps possible, but not as robust, right? Compared to, now tell me about the TG Ovax.
Yeah. So I guess with this, these guys are.
Can I back up for a second? If you assume that what you see, wild type proestrus, and we know, not just our lab, but the field has shown that at proestrus, there's heightened learning,
Mhm.
right? Heightened hippocampus dependent learning. And we know the importance of norepinephrine in the hippocampus for learning, right? So if you assume for a moment that that pattern in wild type proestrus is critical for hippocampus dependent learning, then what would you say about the TG Ovax?
Yeah. Well, I guess with the slower and longer bursts, this was also something that I had a hard time kind of defining because I didn't want to say they were getting less bursty because the bursts were getting longer.
I don't even care about the less, the more. I'm just asking.
As far as that, I would
Yeah.
assume that they would probably show less learning.
Sure,
Because
right?
they're having less
And-
noradrenergic activation, less noradrenaline going to the hippocampus.
And what could you say about proestrus TG by comparison?
I think that they would show more, as far as learning, I think they would show more learning compared to the overactivating animals. Well, I don't know, because this is looking at the direct response to the stimulus. And then this is kind of looking at that, I guess like a longer term. I don't want to say like long term because I feel like there's very specific definitions for that, and this is not But the fact that both of these kind of return to that baseline level, I would assume that they wouldn't show as much learning as the wild type animals.
Right, because of the disease.
Yeah.
But wouldn't you then be able to predict that the TG rat at proestrus, that the ovarian hormones are protective? Because the pattern is more like,
It is.
right?
Yep.
Yes.
I would assume that.
All right, the protection idea was what I wanted to return to in the dissertation, because you you say the word neuroprotective a lot in how you describe estradiol. But in the introduction, at least, none of the sort of canonical neuroprotective lit is in there. It's like, I'm not seeing anything about anti-apoptotic protection against, reactive oxygen species. Improving recovery after stroke, some stuff like that. So
Okay.
I'm interested when you talk about the neuroprotective role of estrogen in this context.
Yeah.
What are you trying to capture with that term?
Yeah, well, so I was thinking about it in this context, what I would assume, and again, assuming that estrogen is beneficial, is that this heightened response during the foot shock, and then this post-shock potentiation that we're seeing is that, it's learning, it's behaving this one way, and it's like, okay, we're gonna keep doing this, because we just went through that, and we're seeing that in the transgenic animals as well. As far as what I was talking about in the literature in my dissertation, the neuroprotective that I'm referring to is specifically looking at animals where they overactimize them, and they either give them hormone replacement back, or they have like a naturally cycling animal, and they see that there is increased dendritic spines, there's more functional connectivity between those neurons, there's increased learning, and so it's definitely more at a top level. And I'm trying to think of any off the top of my head.
Adaptive maybe is a better term for that,
Okay.
or
Well,
proplastic.
synaptic physiologists
Mhm.
would call that protective. It's
Mhm.
protecting that circuit,
It's
not
or
being an antioxidant, although it does do that.
yes, and that's why I asked
It
about
does
that.
do
That's
that.
what I'm looking for, because I know,
Yes.
In terms of a cell biological neuroprotective.
It does
Because
do
to me, what this is telling me that estrogen is really doing is, promoting plasticity, maintaining the ability to have adaptive responses. Which obviously produced, like in the circumstance of responding to stress, that's an adaptive and protective mechanism, right?
Well, maybe.
I mean, at least in the ability to learn faster.
In
That
PTSD.
a specific thing is dangerous. Is helpful, but you're right that in excess it is a problem.
And PTSD is more prevalent in women,
It
so
is.
it might not be a good thing.
Mhm. So then back to Jen's thing of like.
It can be hijacked.
Yeah, for
Yeah.
like all of our favorite stuff is. So yeah, I think I have some comments in there about like, where is the, the distinction between like a developmental cell biologist and a neurophysiologist, that term clearly implies like different.
And in my dissertation, I definitely leaned heavy on the physiology
Mhm.
literature, but
I mean, I saw all the woolly sites. I know all that good hippocampus stuff for
Well,
sure. Yeah.
But this is where a lot of that text, and Steve mentioned this, in your background is not really relevant. Because you don't get to any of that here.
I don't.
And you don't even, when you describe these data, you don't even use those words, right?
So I can take a lot of that out.
Yeah, I think you can.
You don't
simplify.
have
Yeah.
to,
You
but
can
it's
simplify.
not.
I will, and it'll help for the paper. So.
Yeah, for the intro, I
maybe.
was gonna try and direct you to, I thought obviously the summary part of your intro was really well laid out. It covered things in an order that made sense, but that order didn't actually match the way that you had covered things in the individual section.
Okay.
And so my biggest comment for you on the introduction was, go back to that last paragraph and the way that you introduce each of the concepts and make sure that they're ordered thusly.
Okay.
And that also, I think, will flag for you like, oh, I don't say anything, like Steve said, I don't say anything about adrenergic receptors. I don't say anything about estrogen receptors. I don't make a like you say it's ER beta. I do think that's useful when you're talking about in the locus ceruleus. But I think
That was about that section, because I was really trying to pull together that bit, because in the hypothalamus with the GnRH neurons, that they're also estrogen beta dominant, and then also seeing that inhibitory effect similar to what's in the locus coeruleus. And like there hasn't been a ton of work looking in the LC with estrogen. And so like I'm thinking that that could be a similar a similar response to estrogen. But again, like there's not a whole lot of literature in the LC. So I was just kind of piecing that together.
Yeah, I mean, I think there was maybe more than there needed to be, but you're right that you needed to talk about the mechanism of action for estrogen in the LC as far as we know it. And then enough of the other stuff so that you can extrapolate a little bit and make some predictions about what's happening in the LC. So, yeah.
Okay.
Can I ask a question to follow up on Lori's question a little bit?
Yes.
So Lori asked of you about learning. And so you have multiple dimensions of LC activity. You have the baseline level of firing and the burst frequency as well as the burst amplitude, and then also the response to foot shock. You have these different variables. Which of those do you think are related to learning, and how and why?
Yeah, well, I think so. The baseline, I think that that's important for just like understanding spontaneous activity. And I don't think that that is like directly relevant to learning, but I think that that can be like within group, within subject control. I think that the foot shock, like that is definitely an arousing stimulus. And so when one is learning, that is arousal in general. So I think that that can, I don't know, be equated to like a learning circumstance. And then as far as like the post-shock rest, I think that that is kind of suggesting changes in what might be like long-term plasticity. Again, like these are not, I don't know the very specific criteria for long-term plasticity, but then thinking about like long-term learning. So if an animal learns a task one day, that might equate to the foot shock. And then if you retest that animal the next day and the following day, I think that that might be related, or I guess like compared to the post-shock rest. That's how I would think about it.
All right, I wasn't looking for any particular answer. I want to comment though that I just wanted to hear what you thought. One comment though is that baseline activity may actually be important. I think it would be potentially important because it sets an arousal level which can affect learning. You know, there's this arousal performance curve in psychology that seems to be also describe the activity of noradrenaline, where it goes up as you become more aroused, and then eventually you become so aroused you fall off the other end.
You're absolutely right. That is super important.
Interesting.
Right, because you have to have a level of arousal, but then so you can focus attention, right? Because you cannot learn if you cannot focus attention. So you have to have this, baseline level of awareness.
Yeah. So I guess when thinking about it like that, do you think that this baseline, like what you're saying, sounds like kind of sets the stage for the learning that would happen, or I guess the responses happening during the foot shock?
I think it's about the delta.
Yeah.
Well, I think
It does set the stage, but it also I think bursting and tonic firing in my experience are linked. You know, so as the firing rate goes up, the bursts become more prominent, and then as the firing rate continues to go up, they become less prominent because they're drowned out by the level of activity. So there's a neurophysiological manifestation of that arousal curve potentially.
Yeah, and so then you can't detect.
Yeah.
Or you can't distinguish. Did you want to say something, Jim?
well,
Thanks, Steve, for those comments.
I just, we're talking about learning, make sure we're considering Jim McGaugh and Steve Thomas's work. So Jim McGaugh for, the last 4 decades of his career showed that consolidation is, so the 1 to 6 hours after learning requires noradrenaline. So the persistent increase in noradrenaline activity that you're seeing in your first group there might be important for that.
Yeah.
Steve Thomas showed in a Cell paper in 2004 that noradrenergic activity in the hippocampus, dorsal hippocampus, is essential for memory retrieval. And so is it an effect of a loss of memory acquisition, consolidation, or retrieval? Or what's probably more likely, a combination of the three?
Yeah, probably.
So just something to think about. I mean, if you're relating all of this to learning and memory in your paper, definitely make sure that you bring up McGaugh. Probably Steve Thomas's work as well.
Okay, cool. Yeah.
And I don't know if we will in the paper or not, maybe. I just wanted to see how she's thinking about our data and pulling it all together.
Sure.
Because it gets, you guys know this because you do it for a living. You get too immersed in the details. And it's like, what does this mean?
Yeah.
Okay, we got all this pattern, but who
Yeah.
cares,
Mhm.
right?
Yeah.
So, I have one other question for you. What might you be missing in this story
Mhm.
by only recording during the active period?
Ooh, yeah, that's a good question. So something that I'm surprised that no one asked about was what I'm seeing in my transgenic overoptimized animals is this response to foot shock with bursts getting longer and slower. This is the same change that I'm seeing in my light phase units from the diurnal project.
I was trying to get you to bring that up.
Yeah, and I'm glad that you brought it up because like
So now expand on that.
Yeah. So I guess what I was thinking and what I am thinking for the light phase units is that because they're in their inactive period, I think that there are units that this might be a preventative mechanism because like the animal's not, it's inactive. And so by kind of reducing its bursting, that it's not, I don't know, getting overstimulated. It's not hyperexcitable. It's inactive. It's like, nope, we're not doing that. This could be more inhibition coming from that periceruleus. But as far as the transgenic,
I had a completely different thought.
Oh, okay. What's that?
Okay, so, well, you might be right, but I just had a different thought. You just said that is reminiscent of the inactive period.
Yeah, and
So what happens in an inactive period normally?
they're sleeping.
Okay, but this is their active period.
Yeah, well, and we were kind of talking about this the other day, but I mean, in Alzheimer's disease, there's a disruption of circadian rhythms. And so while I'm reporting in the dark phase, I mean, maybe these animals, like they have p-tau on board and now they don't have any ovarian hormones, their circadian rhythms are probably all out of whack. And so who's to say that their locus coeruleus or adrenergic neurons are not operating in their like inactive phase because they're so screwed up. So like that.
Or
could be
they,
it.
yeah,
Mhm. Yeah.
Speaking of the p-tau, you highlight that your model has hyperphosphorylated endogenously expressed tau.
That's
And I
it.
think my memory is that you did walk through the pathway for how you thought the transgenic genes were getting there, but can you remind me
Well,
why you haven't modified tau, but it's hyperphosphorylated in this
so
animal?
in this animal, that pathway that it appears is not completely fleshed out. What I walk through in my dissertation is the hypothesized mechanism for which p-tau accumulates in the LC. And just like the way that p-tau is getting accumulated. I can't remember the exact kinases. I think the one started with
PSK,
that's it, that's
yeah.
it. And that it just becomes hyperphosphorylated because of that. And now I know, at least my understanding of the literature is that the amyloid beta and the p-tau, that they kind of work together in humans. I mean, like the amyloid beta, from my understanding, before there's any like hard molecular pathology that you can quantify in the tissue, soluble amyloid beta increases in the cerebral spinal fluid. Because the locus coeruleus is situated right next to the fourth ventricle, it's going to be right next to that soluble A beta. And so I know that that's one of the hypotheses for why the LC is developing p-tau first. And then thinking about if the p-tau is increasing, or as far as like it's getting phosphorylated because the soluble A beta is up, and then the p-tau is contributing to the A beta and going back and forth. But in this rat model, the mechanism is not known, but I wanted to talk about what's hypothesized to be happening in humans, and potentially that could be happening in these animals too.
So can I ask one last question?
Yeah.
So I thought all of the information that you had contained in your dissertation was largely, again, really scholarly, like you did tons of research, that's super clear. The one thing I struggled with is like how I kind of think about your dissertation and thought about, you know, how this was going to come together was that you are investigating the LC in the context of Alzheimer's disease, right? And like you look at it in your control animals in that first experiment in the diurnal stuff to try to understand sort of normal LC physiology across the light cycle, right?
Yeah.
And then you're looking at it in your OVEX experiment. But you start your introduction with the locus coeruleus. And there's kind of no segue into why we are looking at the locus coeruleus.
Okay.
So one of my suggestions, and you guys can debate how this, I don't think you need to do a ton of like, I think it's just reorganization. I don't know that you need to like rewrite things.
Okay.
But for me, it's like you're going through the LC and you're talking about all this stuff and like p-tau comes up and little bits and pieces of the Alzheimer's disease pieces come into your LC stuff, but then you don't get to the Alzheimer, to the disease model until the very end of your introduction. So I feel like putting that first and introducing the disease and saying that the LC is really important. So here's all this information about the structure and the connectivity and the function and physiology, and like that would make a little more sense.
Okay. And then would I kind of like circle back to Alzheimer's disease when I'm talking about the menopause component of it?
Yes.
Okay.
Yeah, I think it should be like Alzheimer's LC, Alzheimer's menopause, like how it kind of toggle between that. And I don't know the perfect way, but for me, I struggled with knowing what your dissertation was about, but not hearing about it until very late in the introduction. And I think for me, it would make more sense to introduce Alzheimer's and say that the LC is the first place where you see all of this phosphorylation, and that's why we're looking at it.
Yeah. Okay.
So just from the perspective of that, in the intro. And then I have a couple other comments on the document. But one thing for chapter 2, well, I guess it's chapter 3 and 4 then. So chapter 2 with your sort of common methods, and then you go to chapter 3 and chapter 4. And it's like, as I was reading it, I kind of forgot what exactly you did in which part. So I don't want you to be redundant, but there's like, you have little sections in chapter 2 that are like very specific to chapter 3 and then very specific to chapter 4. And I think maybe just put those in those chapters so that it's like, here's all the common methodology. And then here's in chapter 3 and chapter 4, the specific things that I did to look at diurnal, whatever. So I have those comments in here.
Awesome. Thank you.
I think, I like the idea of starting with Alzheimer's disease for sure. I think with Rosie thinking about it, it was hard, right? So she had first, when I got the copy, she had started with menopause. I'm like, why are you starting with menopause? Right? So I said, you're studying the Lucas Aurelius. So talk about the Lucas Aurelius.
And I can totally see that. I think for me, I just struggled knowing that we were going to end with the Alzheimer's model, and that's in her title. And then it was like, I didn't hear about it so long. And so maybe there's a better way.
I agree with you completely. I think if I were writing it, I would have started with Alzheimer's disease. And I think what threw her off was the first project had no Alzheimer's in it.
No.
I was like,
But
relevant.
you can pitch, like you're pitching it as I'm examining the LC to understand how it functions normally, so I understand how it dysfunctions in the model,
I
right?
think so too.
And so I think that's fine.
Yeah.
I don't think you have to, change anything about chapter 3 to make it more Alzheimer-y, but like, so I think everything else was.
I'm telling you, go to 1.5. Your summary and hypothesis, it's like perfectly laid out with all of the same stuff that you're introducing, and it goes like ADLC, like arousal, like it
hormones.
It makes complete sense to me.
Cool. I will most definitely do that.
And I think it's like really just probably mostly a cut and paste situation, and just renumber the.
Well, and that was the last thing that I was doing while I was putting the document together, because like I just could not wrap my head around the best way to organize it.
And I mean,
so I was cutting and pasting, and I was putting things in different.
And I think we talked about it as a committee and went back and forth on ideas and what, and then it's like, and then I read it all and I was like, I think actually I went
the other way instead now. Now that I've seen it.
It's fine going to have 5 of us and 1 of you.
So what do we do should I send like a summary of feedback on the written document to you and you'll collate and share with her? We need to provide whatever our feedback is within 48 hours.
Yeah.
Nice.
Yeah. So, I think maybe it might be, I don't know so I have just all my stuff with like comments on your Word doc. So I don't know if like we just send those to her.
Yeah, that would be the best.
And then you can sort of reconcile between the two.
That would be great, so that I could see like everything in its entirety.
And do you need to see it again?
No, you guys, I don't need to see it again.
I kind of want to see it again.
All right. And she has 2 weeks.
Yep.
3 weeks.
Three weeks.
Oh yeah, 3,
3
that's right.
weeks,
Yeah, so there's plenty of time. You're not gonna need it because it's
Yeah,
like
I don't-
shuffle this and.
And what about future directions? We didn't really quiz her on that. You didn't have any at the end of your talk.
Nope.
Yeah.
I do have it in the document. If you guys want to talk about it, we can. Can I just like, I don't know, bring up some that I'm excited
Yeah.
to talk about? Okay, well, so the obvious one that I did not get to do because of time, I wanted to give my overactimized girls hormone replacement therapy. I think that that would have been awesome, given, you know, the time and the resources to do it. I think that it definitely would have been beneficial for the responses that we're seeing with the foot shock. I think that we would have seen that. And then as far as like the heterogeneous populations, I think that that would have influenced that as well. So yeah, I guess that replacement would have been at proestrus levels, and then being able to see if there was a significant difference between the proestrus groups. So like having another two groups that were overactimized with hormone replacement. And then something that I've been thinking about too is like that critical window for intervention and looking at different time points. Like if we started it right after we overactimized them, I'd assume that it would be similar to the proestrus animals. If we started it a month after that, maybe it would be a little worse. And then if we started it 2 months after that, then it would probably be similar to the overactimized animals. So I think that that would also be really interesting to look at.
There a way to, this is a stupid question, there's not an answer. Or probably the answer is just no. It would be cool to find a way to target these neurons with estrogen in a way that, or in a similar way that estrogen does, that doesn't have the negative side effects of estrogen replacement therapy on the rest of the body.
Yeah, so
Have you thought about that?
yeah, so I guess so you're saying like give the estrogen without the risk of like the endometriosis, the breast cancer that comes. So typically what's done in the clinics is that they'll give estrogen in addition to progesterone, like a low dose of progesterone, and progesterone will mitigate those risk symptoms that come with the estrogen. And so I think that that would be good.
I disagree with that, but
Okay.
based on the literature.
Well, so I think that we would not see as big of a benefit in the brain as uninhibited estrogen, but I think that with progesterone, that would mitigate any like bodily risks, and like that's why they do it in clinics. Pardon?
What about ICV delivery of estrogen?
I-
Limited to, I mean, intracerebral ventricular.
Huh.
I mean, it wouldn't actually limit it because it
Yeah.
would have exchange with the CSF into plasma and eventually get out, but
Yeah,
it would.
be less.
focused,
Yeah,
it would be much less.
Yeah. With that then, I guess like just like from my assumption, I don't think that you would see those bodily symptoms that would come with overexpression of estrogen, but I think that you would still get the benefit
There's been a ton of animal studies where they've done even intrahippocampal delivery. We've done that.
Yeah, and it helps, right?
Yes, yes.
Yeah, but I guess for
But
that, it would be progesterone.
progesterone is really an antagonist of estrogen.
It is. It is.
So you don't see the benefit when you give the two together.
Mm.
Yeah.
This is like real speculative, but just thinking about Jim's question, is there like a way of a formulation of progesterone that like doesn't cross the blood-brain barrier? So like it could have peripheral protective effects, but not central?
Yeah, so
When you stick it in the uterus.
yeah, that's the
You
thing.
can put it in the uterus.
Yeah.
That's where, I mean, the breast cancer is a big risk, but
Mhm.
uterine cancer is a
Yeah.
huge risk.
Yeah.
So you can treat the uterus alone
And
with
progesterone.
progesterone
Yeah.
and then take systemic estradiol.
Yeah.
Because you want estradiol for bone
Bones,
health,
yeah.
for heart,
Yeah.
heart health.
It's been really fun. I've been shadowing with an OBGYN here and specifically sitting in with menopause patients. And so just being able to hear about like what they're going through, the treatments that they're using. Like, I don't know, there've been patients that have come in and be like, I was seeing this doctor, they put me on this, that thing. And then the doctor that I'm seeing, he's like, you don't need any of that. You need this, you need to take a little bit of this. And then hearing women come back and say, I've been on this for 3 months. I'm feeling so much better. I'm thinking so much more clear. I'm much nicer to my husband. And like, that has been really cool.
Well, and you can see, I mean,
Sorry, you said that.
what'd you say?
You get
Be
his wife
nicer
on
to
it.
But, you know, I mean, could be some real signals here with your TGOVAD, because these are young adults and in my experience, young adult, which these are, they can lose our ovarian hormones and do just fine for a long time behaviorally.
Yeah.
It takes a long while with deprivation.
Yeah, and I'm guessing that you're thinking specifically like hippocampal dependent behaviors.
Yes.
And then thinking like, I don't know, from my understanding that the hippocampus is pretty robust. And do you think that, I don't know, that I don't know, with the deficits that would be seen in the locus coeruleus might be more than
It
what's
could be.
seen in the hippocampus, maybe, I don't know.
Yeah, well, there's tons of studies to do,
Yep. Mm-hmm.
because, the whole foggy brain thing.
So if you found after, a century of clinical trials and basic science that estrogen wasn't the way, that there were just too many side effects or something like that, how might you figure out a way to target these cells specifically without affecting the rest of the brain?
Without affecting the rest of the brain. Well, it would have to be something that's specific to noradrenergic cells. And I guess some type of, are you thinking like a genetic model that has some mutation that would only affect the noradrenergic cells? I've never worked with like a Cree-dependent type of.
What about in a human? I'm thinking.
Yeah, he's thinking.
Okay, sorry, can you repeat the question then? Because my brain was somewhere else.
Same thinking about like, if estrogen's not the way to go, but you want to mimic estrogen specific effects on LC neurons.
Yeah.
How would you go about that as a preclinical research to try to inform treatments?
Yeah. Well, I think my girl Maggie brought this question up and we were talking about clonidine. So clonidine also has this inhibitory effect. So I'm presuming that estrogen has this inhibitory effect, but if you didn't want to use estrogen, potentially you could use a low dose of clonidine to kind of mitigate LC activity. I
But
think
that's gonna like affect the whole brain.
Yeah. So then like LC specific, I'm trying to think because the adrenergic receptors that are on it, they're all throughout the body, and the brain. That's a good question.
Yeah. So why are noradrenergic neurons different than every other neuron in the brain?
And not because they produce noradrenaline.
Yeah, why do they produce noradrenaline?
Why do they produce noradrenaline? Ooh, this is a good question. Why do noradrenergic neurons?
As opposed to something else?
Yes, as opposed to dopamine.
I mean, don't they produce a little bit of dopamine?
So why do they
But-
favor noradrenaline?
I'm talking about genetics. They're transcriptionally unique.
Yeah.
So,
Okay.
one could think about, finding transcriptionally unique features of noradrenergic neurons and trying to target those mechanisms
Yeah.
in the future.
Okay.
But I mean,
Do you
Or
know
put a
any
little stimulator in there.
Yeah.
Yeah, do you
that's
know
what I was
any,
thinking.
I guess like transcriptionally unique things about noradrenergic cells? Because that's not something I've spent a lot of time thinking about, but I should.
I don't know anything about noradrenergic neurons.
Okay, yeah.
You can find out.
Yeah, okay.
I'd love it if you guys could figure this out soon, because I can't have ERT and I would very much still like my brain to function. So
Yeah.
like, can we work on this, please? Thank you.
Yes.
Yeah.
Next up.
And the reason I ask is I think that this is a direction that the field is going to go in the next 20 years.
Yes.
Yes.
Yeah, with-
You know.
Single cell, like DNA seq and.
I'll be retired by then, but you can carry on.
Carry on.
I think a lot of labs are going to make
Yeah.
history with
Yeah,
that kind of
which-
stuff.
Yeah, that would be really cool. And then I guess something else that we didn't talk about, but I want to talk about, is I guess like the heterogeneity within the LC, but then also thinking about the peristerelia. So no one asked me about it and I wish that they did, but the 30% and then 18% of cells that I reported that were not sensitive to the clonidine. And so like presuming that those are GABAergic cells, that are out there and like projecting into the LC. But I mean, like, they could be other types of cells. The only literature that I've seen has classified GABAergic cells, but I mean, like, there very well could be other types of cells that are in there. And I guess, like, the way that I'm thinking about it is that, like, the peristerelia seems to be a part of the LC, almost kind of like a shell, and the LC being the core, the noradrenergic core. So then, like, thinking about that too and how that is influencing. Super interesting.
Yeah, this peri-LC stuff is really fascinating to me.
It's super fascinating.
Yeah.
And there's a little bit of data, it's not published yet, but there's a student in the Weinschenker lab that shows that the peristerelis accumulates phosphorylated tau in this model.
Really?
Yep,
How interesting.
I know. And so not only, not only are these,
It's not fake, I mean, it's actually super interesting.
so not only nor adrenergic cells being affected by this p-tau accumulation, the inhibitory regulation that's happening locally on them is also probably affected.
I mean, it'd be interesting to, if you could find a clonidine-like way of seeing if these are the GABA cells.
Right, yeah.
Like a pharmacological.
Yeah.
kind of- that would be cool.
But all
mean,
we have to do is patch on them in the slice
Yeah.
and see if there are modulators that are specific, like, you know, a cholinergic drug or
Yeah.
a something,
Yeah.
a specific metabotropic glutamate receptor.
Yeah.
So like,
Yeah.
you said you haven't looked at the waveforms yet, but that might give you some insight if they all have very similar and, you know, I don't know. I'm thinking about like people who record from the VTA, right? Like there's a mixed bag of cells there, but they're like, we have this waveform that is like the putative
Yep.
dopamine
Yeah.
waveform, right?
Yeah.
So like there might be a
That's
way
even in
to.
like
I-
the context, right?
yeah,
with
I'm
the
sure you're
Zephyr.
right.
And so Steve was talking about this paper that Nelson Toda, I know he's going to be the last author on it, but Michael Kauberman is going to be the first author, and they've taken all in vivo locus coeruleus recordings, and they're like quantifying waveforms, bursting patterns, and so like that is going to be a really good standard to use.
Yeah, there's a few papers.
Yeah.
In here.
Mhm.
Yeah.
And, you know, she still has the other 5 animals to analyze.
Mhm.
There's-
Yeah.
And here's only 4 in each
Mhm.
group. She's got
Yeah,
a lot more
I have like
to
5 more
add.
in each. Yeah, so and that's gonna be super interesting, and now I can do all of these Google Instagrams and bin and file analysis with it, so it's gonna be like a really complete story for sure.
Now you got all the analysis pipelines ready, so it'll be faster.
Yeah.
Yeah, which is great.
Thank you all so much for
Yeah.
helping me train.
Wait, actually, Lori, can I just make one more comment?
Yes.
Oh, so this is quick. Rosie, you have an interesting hypothesis also that rose out of your first data set that there's intrinsic or some kind of mechanism that preserves circadian rhythms in the LC even if the animal's anesthetized. So I think you should unpack that a little bit in the discussion. You should discuss how that might happen. You know, I don't know if I missed it, but I don't think you did.
Yeah, you are right, Steve. I would love her to talk about that too.
Yeah, I think there's no better place to speculate than the last chapter of your thesis.
That's right.
Yeah.
Yeah.
That's a really good point. Thanks for that.
Yeah, and I guess that is something that I've just kind of been thinking about. I haven't spent much time like digging through the literature to look at that, but I can definitely do it.
And as Steve said, you can speculate.
Yeah.
It might work this way, might work that way, right? That's part of the kind of the future directions and how you would go about understanding that more.
Mhm.
And what channels might it be?
Yeah.
Okay, so does anybody else have any questions, or shall we kick Rosie out for a minute so we can discuss amongst ourselves, and then we'll call you back in a second?
Okay.
Stay with me.